Okay. Recording now. I'll throw up while people are coming in, I'll throw up the schedule for today. Maybe just wait another minute or so because I can see people are still coming in. Okay, I'll just start with intro to today, and then I'll introduce our first speaker. So we're going to have, as you all know, we had a previous workshop in October. Kushal and I have been bringing together the experts we know to talk about what happens after GWAS, what kind of methods are available, and then also today getting into whole exome and whole genome sequencing analysis. So previously we heard about heritability, polygenic risk scores, partitioned heritability, and how diverse ancestry plays a role. Today we're going to talk about methods post-GWAS, including TWAS, colocalization, and Mendelian randomization for identifying genes. And then in the second part, burden testing and disease relevant cell types and rare coding. And then the last one is kind of tacked on. Okay, so our first speaker is Dr. William Wen, who's a professor in the Department of Biostatistics at University of Michigan. Methodological interests include Bayesian models, probabilistic graphical models, and in terms of applications, Dr. Wen's interested in statistically sound and efficient solutions to problems in the area of genetics and functional genomics. Thank you very much, and I'll hand it over.
Thank you, Mike. Let me share my screen. All right, so very happy to be here. Today I'll talk about mechanism-aware approaches to implicating putative causal genes, especially using the technique combining TWAS and colocalization analysis. So a little bit background. I think for this audience, this is probably unnecessary, but I think everyone finds GWAS is difficult. Interpreting GWAS results can be frustrating. However, we have to do it because it's provide the key to insights of the molecular mechanisms. What it makes it difficult is because majority of the GWAS hits located in non-coding region. So we don't automatically get what the gene is in those disease etiology. To link causal variants to target gene is a non-trivial task. So there are 2 points. The closest gene are typically not the causal genes. So this is, the paper I cited here is from Open Target, but the study is actually, if you look into it, is come from EQTL study. So if you know the SNP position with respect to the gene, and then you ask the question is this EQTL, the target gene is the closest. So I think majority of the case is not. Not saying this can if you're looking at a different phenotype, may be the case, but the consensus is closest gene are typically not the causal. What makes things harder is you have LD linkage disequilibrium, and then that make everything just more complicated. We'll go through that throughout the talk. So the goal is no longer, when you've done fine mapping, the goal is trying to link those causal genes you find in the fine mapping, supposedly you have high confidence, to putative causal genes. That's not easy, as I said before. One of the advantages now we have molecular phenotype data, especially molecular QTLs. So I'm just trying to focus on a subset of the approaches trying to infer the putative causal genes using these, incorporating these molecular phenotype data. So when we call this mechanism-aware approaches is in comparison to some other approaches you may know, more like machine learning type of approach. One of the main characteristic of the mechanism-aware approach is it does not only provide a target gene, and then if you make that inference, usually you know the molecular mechanism. So the things we're going to talk about, the TWAS and then colocalization, usually link the genes through gene expression phenotype. So there is an explicit link, so we call that mechanism-aware approach. I'm going to focus on the gene expression throughout the presentation, but these methodologies can be applied to other molecular phenotypes. So there are two types approaches I'm going to try to cover today. One is colocalization. So the definition is pretty simple, but there is emphasis. So identify the genetic variants that causal to both gene expressions and then complex traits. I'm throwing the word causal there because statistical 101 correlation is not causation. In this case, it can be interpreted that way because Mendelian inheritance is a nature-made randomization experiment. So if you find genetic association and then you pinpoint that genetic variance, usually the association is interpreted as a causal variant. So the colocalization analysis has a high standard. They want, at least the goal is trying to overlap the trait variance at the SNP level, at the highest precision. That makes the thing harder. The second type is transcriptome-wide, should be wide, association analysis. So this is trying to detect, so this is a definition based on the procedure. So what they do is detect association between genetically imputed gene expression and then the complex trait. It doesn't really explain why the TWAS provide you causal genes or putative causal genes. Some of the reasoning I'm going to explain, and then you're going to hear Jean talk about Mendelian randomization for the second half of the hour. So they're all related. So let's focus first on the colocalization analysis. So what we're trying to do is confidently identify causal variants from GWAS. Well, I mean, those overlappings. However, the biggest hurdle is, we all know, confidently identifying causal variants from GWAS is intrinsically difficult due to LD. So this is a study from 2016 from Mark McCarthy's group. They have quantified the lead SNP from trait association. So those are common SNPs, are often unreliable. So they show there is a spectrum. In your analysis, you're using your best available fine mapping approach. You pick the lead SNP. So they find that the lead SNP is truly causal SNP. It really depends on the effect size. If you have effect size like log odds ratio 1.1, it's about 5% of your lead SNP eventually turn into the true causal SNP in the simulation setting. They are using the true genetic genotype data from, so that represents the real LD here. So the difficulty is because of LD. And then if you cannot use a lead SNP to represent the causal SNP, then the second best thing you could do is using a probabilistic quantification, right? So there's many advantages using a probability to quantify if a particular SNP is a causal or not. Because some of the complex scenario we're going to encounter in this type of colocalization analysis, you can imagine there are two perfectly linked SNP, and then you just know one of them is causal to the trait of interest. So you're absolutely sure one of them is the causal SNP, but you really have the maximum uncertainty on which one is the true causal SNP. That's fundamentally important for colocalization. So the way you could do is assign 50-50% posterior probability to the two SNPs. Then you can, they create a lot of, you have comprehensively characterize this rather complicated scenario. Okay, so as I said, the causality of the SNP are characterized by their posterior inclusion probabilities. So there's a carry over the uncertainty into the colocalization analysis. So here I'm only trying to emphasize the gamma here is a latent indicator. So if gamma equals 1, that means the particular SNP I for the trait T is a causal SNP 0. Otherwise, that means it's not a causal SNP. Okay, so we get the probability from the fine mapping. And that has been the common practice. I want to say a few. I think people tried using standard hypothesis testing and then using likelihood-based approach for the similar reasons I cited before. It's not as effective as the probabilistic approach. And then when you use a probabilistic approach, is the best way to do that is within the Bayesian framework. Okay, so for any colocalization analysis, you can formulating the problem as just trying to find this joint posterior probability. So evaluate. So the E here represented, so you have a SNP I is causal for both trait E representing expression and then trait G representing a GWAS. And given the GWAS data and the EQTL data. So your input is typically the fine mapped GWAS data and EQTL data. It's on the first line, just the PIPs. So I'm not going to go through the details of this calculation. It's a standard Bayes rule type of the calculation, but you do need some prior information on the enrichment. So this is written as a conditional probability given. So the interpretation is very straightforward. Given the SNP is EQTL, what is probability being a GWAS hit? So you often read this type of research, at least like 10 years ago. There's a paper cited like thousands of times. The EQTL SNPs are more likely to be GWAS, or the GWAS SNPs turn more likely to be a EQTL, saying that this probability is probably, so this two random variable, the gamma GI and the gamma EI, so those are the causal status, latent. These two things are not independent from the data we have observed. So, so evaluating these, or how to set this value, becomes kind of a distinction factors in different colocalization analysis. So there's some of the popular ones, the eCaviar. So they do assume a priori independence. So the gamma. So in that case, it's very simple. You just multiply the two marginal fine mapping PIP, you get the eCaviar posterior probability for colocalization. So you can imagine this very conservative. So the colloc is a package been highly cited, highly used. So they allow user to specify that value. And then the fasting log. Our group developed this. I think with everything else being standard Bayesian, so we're trying to estimate this enrichment probability from data. So it's kind of an empirical based type of the framework. So I have to say, so those enrichment parameters is highly sensitive to the result is highly sensitive to the prior. So multiple authors has shown that. So if you have the data, obviously my recommendation is estimate that prior, at least justify the prior, understand what the particular prior you put in to your analysis, what that really means, if the enrichment level unrealistic or not. So even when enrichment priors are properly set, we show that colocalization analysis really depends on the quality of the fine mapping. This is easy to understand. So if you cannot marginally do a good job find the causal variance, how could it possible you find the colocalization in high quality, right? So usually it's the garbage in, you don't expect anything else out. And even you have the good data there, but there's overly conservative with high specificity and a low sensitivity. So all of these are related to, because due to the LD, and then we cannot confidently identify the causal variance in a single trait, in any traits, so to speak. So there are two sources of power loss. One is one of the marginal analysis completely miss one of the causal variants. In that case, you won't see colocalization. The other source of the power loss are slightly different. We call it false negative arrows. Is they all detect the locus correctly, but assign the probability different to different SNPs. So your marginal analysis telling you, although the causal variance is in the same locus, but they don't necessarily give you the evidence they are on the same SNP. So those things as input for colocalization will affect the power or the sensitivity of the result. All right, switch gear to the TWAS. As I said before, the TWAS is not, well, I mean, in common practice is defined by its procedure. So what everybody does is they're trying to predict the so-called genetically imputed gene expression level, and then do the association. But the reason is you can claim causality from TWAS finding is really related to instrumental variable analysis, Mendelian randomization analysis. And then it's rightfully so, I think, among other competing causal inferences approaches like mediation analysis, I think applied TWAS or MR in this context has unique advantage. So although there's assumptions associated with every analysis, but at least the TWAS MR type of analysis acknowledge unknown confoundings. And then this is critically important when you're trying to infer causal genes. You cannot just say, I don't consider any confoundings. And then the difference between the TWAS and then the MR maybe is its emphasis on testing rather than estimation. At least in this stage, at the stage you're trying to try to discover the causal genes. So this is the graph. I think Jim probably gonna talk more about it. So you have a null model set up. So the G is the genetic variance associated with X. That's the gene expression. So the G is EQTL. But if there is no arrows linking from X to Y, so there's no gene to trait effect is exactly 0, then you have a null model. So the alternative model, the causal model, is you put back into that link. Noticeably, there is some other links missing. So this is a common assumptions behind MR. But this type of you have a causal model and a null model, then the TWAS procedure can be effectively distinguish the null model from the causal model, or the causal model from the null model that way. So to test the causal link of the gene, so you compute a weighted combination of your instruments, the genotype of the instruments. So this is interpreted as predicted gene causal predicted gene expressions, and this is also related to the well-established two-stage least square algorithm they also call for. So this is just trying to write the graphical model into a sequence of linear equations. So the M here is, is the expression level. So the G here is, all these, the EQTLs. And then Y is the complex trace of interest. So the gamma is the gene to trait effect. So if you want to assert gene to trait effect not equal to 0 under the assumption of the MR and the correlation, you can sufficiently show that by showing this predicted or this linear combination of the instruments is correlated with Y. So simple as that. I think the importance is really understand where the causality implication come from. There's most of the TWAS paper, at least the one they wrote, they trying to stay away from the causality claim, but people use that anyway. So the available approach, there are too many to cite, but they're usually different in the weight construction. How do you combine the instruments? But they're similar in performance. And then we're just trying to talk one of the biggest caveats from the not surprising, this is also come from LD. So trying to speed up to give Jin more time on the MRs. But so the idea here, the LD can screw up the TWAS. So typical thing is if you run the TWAS for any trace, you find a lot of signals, because this is a complete opposite to the colocalization analysis. This is probably the reason people tend to use TWAS more than colocalization in a way. But noted there are a lot of spurious, at least PCGs. So those signals are real. The association signals are real, but the interpretation of they are following the Mendelian randomization assumptions, therefore they are causal. The causality can be invalidated. So the simple case of that is just LD hitchhiking. We call this. So you can imagine if there is a causal SNP, a causal EQTL SNP, and then even modest LD with a causal GWAS SNP, they are not necessarily overlapping. And then what you can see is the TWAS score is driven up by this potential LD. So in theory, that leads to the violation of inclusion, exclusion restriction. That's violating of these. So the figure here showing the panel A is a real data example. So we're looking at the UK biobank standing heights. This is on chromosome 3. So you get a bunch of TWAS significant signals around this position, 141 megabase. This whole thing is driven by a single GWAS signal. So look at the nearby, especially the nearby, the signals. It's all high up. It's not as high as this one, but it's all high up, right? So we are managed to use a simulation to just using the real genotype data and then specify the causal GWAS to recreate this pattern. Okay, so you have seeing this B is similar to A, but once we control, we regress out the GWAS effect for that lead SNP, high SNP. So all signals got disappeared. So that means that proves this everything here is just hitchhiking. Doesn't mean they have causal effect. As a matter of fact, we're not even considering a causal model doing the simulation. So all these are sort of artifact. I think I can just wrap up very quickly. So the solution to that, I think, is you observe TWAS has high sensitivity but low specificity. And then colocalization is in the opposite spectrum is have high specificity but low sensitivity. Really, the true colocalization analysis is quite reliable, especially when you set the prior correctly. But if you look at the causal model, so there is a natural thought to combine the two approaches together. And then the motivating thought can actually come from if you look into this structural equation, and then if you want to do substitution of m from the first equation to the second equation, you will realize all the causal EQTLs have to have non-zero effect to the trait, meaning they are also causal GWAS, at least for this model. So that gave you a sort of a necessary condition saying, if you're looking for the true TWAS satisfy this diagram or satisfy this the causal model, then necessarily the colocalization is one of the necessary condition for the true causal genes. So for PCG's implication. So we just implement this idea. It's our recent work. It's very simple within the Bayesian framework. Just treat the colocalization evidence as a prior. Same idea as you using lasso or ridge regressions. You use prior to implement some sort of a restrictions, right? So the constraints on the TWAS, and then use that essentially as a filter to the TWAS result. And then it has pretty good results in practice and in simulation. I won't go over for those. So I did not plan to show this, but I think there's one of interesting use case scenario. So this is an analysis of 4 molecular trait GWAS data from UK Biobank. So firstly done by, well, I don't mean they are the group, but Johnson Pritchard's lab did some of these analysis to showcase the omnigenics model. So we analyze their data using this by incorporating the GTAC EQTL data. So there are some interesting findings. And more importantly, I think that the message is, if you have probabilistic quantification of PCGs, they are actually allow you to do more efficient and rigorous gene set pathway analysis, therefore discover more things. So all these are in the preprint. I think this is some of the citations I have for the paper. And then thank you very much. And yeah, I'm happy to take questions if I have time.
Thank you so much. Yeah, I think we do have time. We started a couple minutes late, so why don't we take some questions now, and we can switch over in like a couple minutes to Gene.
Okay. I didn't see your message, by the way, sorry.
Yeah. Andrew, do you want to go first?
Yeah, thanks, Mike. So that was awesome, and it's going to be, I think, a great resource for the consortium. And I'm curious about the colocalization in TWAS. It seems like the colocalizations, all the methods seem to be more Bayesian driven. And I think that makes sense given the problem. Well, the TWAS is almost like frequentist, which is like a weighted linear combination. I'm curious, has there been any thoughts of trying to do like the reverse in either like TWAS approaches, like using Bayesian statistical approaches, and then using colocalizations using frequentist type thinking?
Right. Good question. I think we tried. I think the TWAS part, at least there is a Bayesian components in it. I think in terms of constructing the weights, there's a lot of Bayesian approaches, including ours, is trying to using because we're not certain which is the best predictive model. So using a Bayesian model averaging sort of idea to average overall. So, and then I know there's from Emory, I think it's from Young's group, they have called a Tiger or something. It's a Bayesian approach for TWAS. So there are efforts on that, but I think the issue is, I think we don't see a problem with frequentist method. Yes, Bayesian can improve the performance potentially, but not a lot. So the focus has been like correct for this LD hijacking, correct for this potential violation in horizontal pleiotropy. For the colocalization, it's really hard to formulate as a frequentist approach. Thinking about testing, so you have a null that's saying it's not colocalized, but it's a composite null. There's a lot of scenarios you need to cover. So it's almost impossible to, it's difficult, if not impossible, to come up with a p-value for that sort of a testing. I guess that's why I think combined with, people think more about carrying over uncertainty. So the Bayesian is kind of a natural to their solution.
Yeah, I guess one quick follow-up is like the intact method, which I think was really nicely motivated at the end. Is that, so would you describe that as sort of a, yeah, it is a Bayesian approach. Okay.
Right, but we take the z-score. So basically we turn the z-score, the association z-score, that can be frequentist, a Bayesian or anything. So that turn that into a base factor. So that's
okay. Thank you. Appreciate it.
Yeah, maybe one more question. Kushal, you have a question?
Yeah, so yeah, great talk, William. So one question I had was that, so there is this TWAS fine mapping work from Bogdan and Nick Mancuso's group that also corrects for the LD in the TWAS association results in general. So I was just wondering if you have compared your results with that, or how it,
yes, we did. Yes. I think so the difference is, I think their idea is valid. So in a way, we should treat TWAS like GWAS. So each data points, especially in this plot, is a gene, but you see the similar pattern, right? So you see a cluster of, in the GWAS, you see a cluster of variants get together because of LD. And then their idea is just using the, the fine mapping idea to sort it out, which one is the gene. So it's effective in this way. We call this between gene LD hijacking, but you can imagine the LD hijacking can occur within the gene. So they're just too nearby. And then, so that method is a little bit vulnerable to this type of the situation. On the other hand, if you're just simply put some sort of a colocalization criteria prior in there, they very effective to remove. Actually, the best case scenario is use both. I can talk to you about that afterwards, but yes, I think the two sorts of idea are independent. You can combine them.
That's great. Thank you.
Great. So I'm gonna just quickly introduce Dr. Jean Morrison. He's assistant professor in the Department of Biostatistics at the University of Michigan. Interested in statistical methods motivated by problems in genetics and genomics, understanding patterns of pleiotropy across a large number of traits, and also interested in Bayesian methods, causal inference, and robust and non-parametric approaches. So a perfect speaker for today. Please, Jean.
Thanks. Hi, everybody. I will share. We'll do the awkward thing where I share and then I ask you if you can see it and go to full screen. So okay, hopefully that looks like a slide to everyone. Yell if it doesn't.
Looks
so I'm
good.
going to talk about Mendelian randomization, and I'll just preface this by saying, at least for me, this is a very dense topic. If you take my class, I make you learn about Mendelian randomization for like 2.5 solid weeks out of a 16-week class. And that's, I feel like maybe even also not enough time. So this is going to be sort of an overview. There'll be quite a few references to come back, and I'm happy to answer questions. So usually I start with a bunch of these causal graphs that William showed, but I'm going to start with maybe some intuition, and then we'll see some causal graphs. So here, this is a like stretched out and zoomed in version of, maybe you've heard this called a Miami plot, which I think is sort of a clever name because you have maybe the city reflected in the water. So, so on the top is a Manhattan plot for LDL cholesterol, and we see all the all the peaks, all the variants that are genome-wide significant popping up above that dashed red line. And then on the bottom is a GWAS for coronary artery disease. And so the thing that I want to draw your attention to is if I look at my peaks on the top, almost all of them are mirrored by a peak on the bottom. And maybe it's a little bit smaller, right? So in this case, we have, can you guys see my mouse, hopefully? So
Yes.
if we look like on chromosome 17, right, there's some peaks over here, and it looks like there's sort of mirror image peaks on the bottom, but they're a little bit smaller. And most of these LDL peaks we can match with at least something that's sort of close to genome-wide significant on the bottom plot. And maybe this peak on chromosome 5 is an exception. And that's sort of the motivating place, right? If we do this GWAS and we say, oh, this LDL cholesterol is sharing so many variants with coronary artery disease, there must be a connection here, right? And Mendelian randomization is sort of about formalizing this intuition of these two traits are sharing lots of their variants, and what does that mean? And so one thing I can do here is I can go through, I can scoop up all of my genome-wide significant LDL cholesterol variants, prune them for LD so I don't get multiple representatives of the same region, and I put them on a plot. And so this is kind of a classic Mendelian randomization type plot, and in a couple slides you'll understand why. So on the horizontal axis, I've plotted the association with LDL cholesterol, and then on the vertical axis, I've plotted the association with coronary artery disease. And so this plot kind of confirms what we can see looking at the Miami plot, that each of those peaks really is mirrored by an association with coronary artery disease. And it looks like there's kind of this positive association, right? Variants that increase your LDL cholesterol more seem to also be associated with higher increases in risk in coronary artery disease. And this is sort of pushing us towards thinking, well, that kind of seems consistent with the idea that LDL cholesterol is a risk factor. LDL cholesterol is increasing the risk of coronary artery disease. It sort of supports this hypothesis that LDL is mediating the effects of these variants on CAD. So starting with that intuition, now we can like formalize why we think that's true in more of a formal statistical way. So the idea that connects that intuition with actually being able to estimate a causal effect is this idea of an instrumental variable. So William drew this directed acyclic graph, this DAG, and I will now draw it again. You'll see this graph a couple of times. So the idea is, let's say I have some variable, which I'll suggestively call GJ, and you don't yet know why I've called it that. And I know that this variant causes my exposure. So my exposure here might be LDL. And I know that this variant is not linked functionally to my outcome in any way that's not mediated by X. So these, formally, I can write these as these assumptions. So GJ causally affects X, which is called relevance. I'm giving all of these terms. So these are like the terms that people use for this in instrumental variable analysis, in like econometrics. So we've kind of, we talk about the same things with different words. And so I think it's nice to have the like translation between fields so that you can read papers from both fields. So assumption 2 says there's no common causes of GJ and X and GJ and Y. So if I have confounders of X and Y, they don't affect GJ. And then the final assumption says that GJ can't affect Y through other pathways. So that's these orange arrows. So I used orange for pathways that aren't there and blue for pathways that are there. And the intuition here is that if GJ satisfies these 3 assumptions, then it is behaving exactly like a random treatment assignment in an RCT. So if you think about an RCT, you've randomized people to groups. And what happens? Well, that group determines their treatment. So that's A1. Group determines the treatment. I know that the random group assignment causes the exposure. And then the other important thing about a randomized treatment assignment is that it's independent of all of their confounders, and the randomized treatment assignment doesn't in some other way affect their outcome except through the exposure, right? The random treatment assignment only determines what treatment you get. It doesn't determine anything else. So that's why we believe estimates from an RCT. And this graph is saying the reason we believe those estimates is because we have these three assumptions, and any variable that satisfies those three assumptions is just as good. So it doesn't have to be a random treatment assignment. I could go out into the world and find a natural instrument, and it's going to be just as good as a random treatment assignment in an RCT for measuring my causal effect beta. So Mendelian randomization is just, I'm sorry, this should say IVA, so in the title. So it's just instrumental variable analysis with genetic variants as instruments. That's the whole thing. We've just come up with two different words to mean the same type of analysis. There are differences between the kind of analysis that we do in MR versus the kind of analysis that economists are often thinking about when they do IVA, but nuts and bolts, it's the same concepts. So if the of the three assumptions, right? So now we just need to know, can I verify that a genetic variant really is a good instrument? So of the three assumptions, we can test the first one, right? So I can find out, is my potential instrument at least associated with X with a linear regression? The reason that we like genetic variants as instruments is because I can go ahead and just rule out a lot of violations of the second assumption. So the second assumption was no common causes of the instrument in X or the instrument in Y. And because genetic variants are fixed, I think, okay, your air pollution exposure is not going to change your genetics. Your medication that you take isn't going to change your genetics. I can rule out all of those incoming arrows. And so that makes us very happy. And we think this is going to be a good instrument. And then the last step is just, well, I got to justify the third assumption, right? The exclusion restriction. And this is going to be like the sticking point and the thing that we spend a lot of effort trying to do in MR. Okay, before I get into how to actually do MR, there's this maybe divide between, am I going to do my Mendelian randomization with summary statistics or with individual level data? So I'm going to be talking primarily about summary statistics. I'm going to talk only about summary statistics for the next 15, 20 minutes. And the reason for this is summary statistics are abundant and they're small compared to individual level data. So if I've done a GWAS for my favorite trait, and now I want to know, does vitamin D levels cause my favorite trait? I can go into this database and just download summary statistics for vitamin D levels and do my analysis. It's actually very, very accessible and very, very easy to do. I don't need an IRB to go get GWAS summary statistics. If I want individual level data, I need to either collect it or, there's more regulatory oversight, and it's huge potentially. So there's some trickier issues with using individual level data, but you can do a little bit more with individual level data than you can do with summary statistics. These are just a couple, I think, interesting individual level data extensions that if you're interested in this, you might want to go learn more about. So if you want to estimate nonlinear causal effects, there are some methods for doing that. There are also some clever methods for correcting for violations of assumptions if you have individual level data. I like this almost exact MR paper that came out really recently where they use parent-child relationships and model the meiosis process. So that's just some stuff that you could look into if you find it interesting. Okay, so let's say we want to do summary statistic MR. Our outline of our workflow, before I tell you how to do each of these steps, is step one, we got to identify some instruments for our exposure. And how we do this is going to depend on what estimation method we want to use later. Step two, I'm going to extract those associations from the outcome GWAS. So I need to know associations in exposure and associations in the outcome. Step three is important. I need to harmonize the data. So I need to make sure that the effect alleles match up. If we don't do that, we get the wrong estimate. Step four, typically people don't just pick one estimation method, and we'll see sort of some of why that is. But typically you're going to apply usually at least 2 methods that make different assumptions, and this is part of making sure that you have effect that is robust to changes in the assumptions that you make. And then step 5 is some form of assessing the robustness of your results to assumptions, checking your evidences, checking for evidence of violations as much as you can. So all of these steps, well, step 2 and 3 are fairly straightforward. Steps 1, 4, and 5 definitely have nuance. I don't have time in this talk to go into all of the nuance, but I'm hoping you should have in your mind that these steps have important choices in them. And so understanding what's going on there is important for getting a good causal estimate out of your MR analysis. So a little bit of notation before we get into some of the MR methods. So we're going to use this big gamma, little gamma notation, which you'll see across the MR literature. So big gamma is going to be the association of GJ with Y, and then big gamma hat is the estimate of that association, and this is what comes out of GWAS. So in GWAS, we do a regression of GJ on Y, and I get out big gamma hat, and then I should also get out an estimate of the variance of big gamma hat. And that's what I mean when I say summary statistics. That's what's going in as the input data. And then on the other side, the little gamma is the association of GJ with X, the exposure. Okay, so the sort of baseline simplest strategy for estimating this causal effect is called inverse variance weighted regression. And what's going on here is I know if all of my assumptions are satisfied, then I can say, well, the expected value of big gamma hat, which is just big gamma, is beta times little gamma. Because we know that that variant can't affect the outcome through any pathway besides X, beta being the average causal linear effect. We have this relationship in the regression-based approaches. Generally, the strategy is, well, this formula includes little gamma, which I don't know, but I do have an estimate for little gamma, which is little gamma hat. I'm just going to plug that in. So instead of fitting this regression on the top, which is sort of infeasible because I don't know little gamma, I'm going to fit this regression on the bottom, where I've just plugged in little gamma hat. And I'm going to weight by the inverse of the variance of big gamma hat. So I'm essentially just ignoring the fact that I have measurement error in little gamma hat. One thing to keep in mind about this approach and most approaches, unless they advertise otherwise, is that they assume that all of the instruments are independent. So I have to prune for LD before I get started. Okay, so weak instrument bias is a common problem that you'll hear talked about in MR world. And formally what this is finite sample bias in our estimators. So they're going to be biased either towards 0 if there's no overlap in the GWAS samples, or towards the observational association if there is sample overlap, which is a problem because the whole point of doing this is that we didn't want an estimate that was biased towards the observational association. So asymptotically, our estimate is going to get close to the true causal effect, but we always live in finite samples, and so we care about that finite sample bias. So important thing to know about IVW is that the size of the bias is proportional to the inverse. We need inverse in here somewhere. The inverse of the average instrument strength. So the larger the average instrument strength, which means like average expected z-score, the less bias. So if we want less bias, we need to choose instruments that have large z-scores or small p-values. So a typical thing is to use a p-value threshold of 5 × 10⁻⁸. If you do this selecting in sample, you end up with a small amount of selection bias, although that I think is about an order of magnitude less than the finite sample bias that you'll get anyway. So the the formally best thing to do is really a 3-sample MR where you select your instruments using an independent GWAS for X. So this alternative to the regression approach is this likelihood-based approach. And I think most of the like maybe modern fancier methods are in this likelihood-based formulation. And so in this formulation, we have this, we say, all right, we want to admit that there's some measurement error on little gamma hat. And so I have this bivariate normal distribution for big gamma and little gamma. This is the same model that we were considering when we proposed the regression approach. The only difference is that I have this sigma j, where I'm admitting that I have measurement error on little gamma hat. So how am I going to estimate beta once I have this model? There's a couple of different options. So Limmel is from 1949, and in this strategy, we maximize a profiled likelihood. And then there's also there's variations of Limmel that are better behaved in some ways that I'll talk about coming up. There's also Bayesian options, right? So the big problem here is that I have these nuisance parameters, little gamma. So one option is I can put a prior on these nuisance parameters and then get a posterior for beta. Almost all methods also assume independent variance here. Okay, so these methods still experience some finite sample bias. So this is, I think, kind of a unique feature of Mendelian randomization estimators, is that they pretty much always have some small amount of finite sample bias. The important thing here, so in Limmel, that finite sample bias is proportional to the inverse of the total instrument strength, which is much better than being proportional to the average instrument strength. That means that we can afford to include some weaker instruments as long as we have strong instruments in there. All right, so that's baseline methods that work if all of the assumptions are satisfied. What do we do if the assumptions aren't satisfied? So there's this term horizontal pleiotropy, and this horizontal pleiotropy is a very long word that means is equivalent to saying violation of the exclusion restriction, which is also long. Statisticians are not great at naming things. There are two flavors of horizontal pleiotropy, and I'll sort of try to convince you that it's the difference between these flavors is important. So what I'll call uncorrelated, or maybe you would call it sporadic pleiotropy, this is what arises when GJ is associated with Y, but not through some pathway that's shared by a lot of the other variants. So this might happen due to the LD that William was talking about, right? So when you have a causal variant for Y and it's right next to a causal variant for X, there's LD between them. We end up with some sporadic horizontal pleiotropy there. The other flavor is what I'll call correlated horizontal pleiotropy, and this occurs when instruments for X are actually affecting a heritable confounder of X and Y. So we had previously assumed, well, our instruments are maybe independent of all of our confounders, but we know that many things are actually heritable, right? So behaviors that people might have or other molecular risk factors could have their own variants. If some of our instruments are actually affecting X through a confounder like U, we're going to have a horizontal pleiotropic effect, and importantly, this effect is going to be correlated with the effect on X, and that's why it's called correlated horizontal pleiotropy. So how do we account for horizontal pleiotropy? This has been like the big project of MR methods. I'm not going to be able to summarize it completely in this slide, but I tried to give an overview of what people have done, right? So the basic idea is I've had this mean model, gamma j equals beta little gamma j. What I need to do is I need another term in there, I need alpha, and then I need to say something about alpha, right? What's the distribution of alpha? What's the mean of alpha in order to fit the model? And all of these methods differ from each other in essentially what they assume about alpha. So Egger regression basically just fits IVW with an intercept. It assumes that alpha is either constant or is independent of gamma j. There's some other variations of the regression method, the median and the mode, that try to just be resilient to having large alphas. And then there are likelihood methods. So the MR-RAPS method is an extension of that limbal method where you add in an alpha that's normally distributed. And then finally, there's these mixture models. So I'm going to talk a little bit about the mixture models. I developed one of them, and so I have some investment in the mixture models. But I think they're also a good idea in this setting. So there's cause, MR-aid, MR mix, and a few other likelihood-based methods all use a mixture model for alpha. So what does the mixture model for alpha look like? So it says, all right, I think that some small proportion of my variants exhibit correlated pleiotropy. If all of my variants exhibit correlated pleiotropy, I have an identifiability issue and I'll never be able to separate the causal effect from the confounding effect. But if it's just a small proportion, I should be able to model something. So the way we'll do this is we'll say gamma J, this is this term that we've always had, this beta little gamma, and then we'll have theta for this will be like our sporadic pleiotropy. And then this last term, ZJ, eta, gamma J, and I'll talk about what that means in a minute. So theta J is our sporadic pleiotropy. So it's just got some normal distribution that's the same term that MR-RAPS was adding in. ZJ is this indicator variable. So ZJ is 1 if we're a correlated horizontal pleiotropy variant, mouthful, it's 0 otherwise. And if we're in this case, then we have this additional effect, which is eta times gamma J. So this term is allowing for us to have some horizontal pleiotropy that's correlated with the direct effects. So the other term that you'll see for that assumption is the inside assumption says that the pleiotropic effects are independent of the direct effects. And this is saying we don't need to have the inside assumption. We can have some violation of that. One way of thinking about this is to say that we have a single heritable confounder and some proportion of the variants that are instruments per X are acting through that heritable confounder. So this mixture model is used by a bunch of different methods. So in cause, we use a Bayesian approach. So we start with this mixture model, and then we end up computing posterior distributions for all of the important parameters in here for pi and for eta and for sigma squared theta. And then we just compare, do the posteriors fit the data better in the causal model or in the non-causal model? And that's sort of your answer for whether or not there's a causal effect. There are frequentist alternatives to this. So this is a few of them. MR-aid has the same mixture model. It also allows for correlation between instruments, which I think is really nice, especially if you have a very dense genetic architecture, right? So if a lot of your causal variants are in the same region, this might be advantageous. It uses a Gibbs sampler. So we've had a little bit of issue with the Gibbs sampler not converging, but I think that's overcomable by fiddling with the parameters. MR-APS is a new method that I think looks cool that I haven't tried out, but I really like the model. So they also have some potential, they have this foreground background model where they account for some potential confounding in GWAS estimates. MR-CML is another one, just like a little overview. So cause tends to be, I think, the most conservative of these methods. It seems like MR-CML and MR-mix might be a little anti-conservative, but don't pin me down on that one. That's just my current impressions from looking at results from different papers. And then before, I think we're slightly over, but I'm a couple slides from the end. So the last thing I wanted to talk about is multivariable MR, because I think this is a really exciting field. So in multivariable MR, we're trying to estimate multiple causal effects of multiple exposures at the same time. And this is going to help you if you're interested in mediation proportion, or if you're interested in directly controlling for one of these heritable confounders that you have measured, right? So now I need GWAS data for this heritable confounder. if that's the case, I can use multivariable MR. So there's multivariable MR, multivariable extensions of both regression and likelihood strategies. They both have some limitations right now. And so this is sort of a field that I'm very excited about. I'm working on a method that will hopefully be out in the near future that should be useful for overcoming some of these limitations. So multivariable IVW really doesn't perform very well in large models. And when I say large models, I mean really more than 4 or 5 exposures at a time. And this likelihood alternative does do well, but it's tends to not converge in its attempt to maximize the likelihood in larger models. So that's sort of the problem that we're working on there. I wanted to include some review or overview papers that might be useful if you're thinking about applying MR or if you're working on MR methods. This third one is sort of more statistically technical. The second one is gives some like very directed advice about how to do each of the steps. And then the last one is this MVMR review, which I think is nice. And then I also wanted to give some resources. So if you are doing Mendelian randomization, you probably need a nice source of GWAS summary statistics. So many of you maybe already know about these, but I think it's nice to have the links. So the MRC IEU database it's very nice. Everything is in like a common format. There's some like summary data about the different studies. The NHGRI catalog is also getting like increasingly standardized and is nice. They both have APIs. And then there are some useful R packages for IEU GWAS R can be used to query the MRC IEU database. And then 2-sample MR is a really useful MR R package. So that's resources. And then if there's time for questions, I'm happy to take questions.
Thanks, Jean, for the very nice introduction to MR. Maybe we have time for one question. Maybe I can ask a question. So we have been very interested in different types of QTL data as part of the IGVF. And I just wanted to know your thoughts on how to handle like chromatin accessible QTL or anti-QTL data together when we have that under the MR scenario.
Yeah, so something that we've been looking at is building networks out of these. So one issue with QTL data, if you're looking at something like gene expression, right, you are in this state of having like maybe a more dense genetic architecture, which tends to make MR and TWAS not very different because you actually don't have too many different independent biological things happening, right? So even if you have multiple causal variants, if they're all kind of acting through the same biological process, you kind of have less information there. You can still apply the same MR techniques. There's some limitations about like the extent to which you can check your assumptions, right? So when you check like heterogeneity assumptions, you might not really pick up on it, the amount of horizontal pleiotropy that's there because all of the variants share the same horizontal pleiotropy. I think MR does best when you have multiple biological pathways contributing to an exposure, which does happen, right, in some of our QTL data. And really like the nicest situation, which you can start to get out with annotating these variants, is when you can say that you sort of, you know something about why this variant is affecting your exposure, right? It's like regulating an enhancer, or it's coding variant or something, and you can actually like select your variants in a more bespoke way rather than a more shotgun way. There's kind of a trade-off between accuracy and efficiency in the like, bespoke MR versus shotgun MR, right?
Great, Zane, thanks so much for the talk. I'll probably move on to the next speaker. We have Zilin Li. He's an assistant professor in the Department of Biostatistics and Health Data Science at the Indiana University School of Medicine. And he is interested in statistical genetics and high dimensional data applications. And today he's going to talk about rare variant burden tests.
Thanks for your introduction, Dr. Day. Good afternoon, everyone. Thanks for attending my presentation. Today, I'm going to talk about why do we do burden testing. Let me start with an outline of my presentation. First, I will give you a background of the large-scale whole genome sequencing studies and its statistical challenges, especially for the rare variant association analysis. Next, I will introduce STARR pipeline, an all-in-one rare variant analysis tool for biobank scale whole genome sequencing studies. And then I will apply a STARR pipeline to the TOPMed freeze-5 lipid data and to give you an example of the real data application. And finally, it is the conclusion. For every one of us, each human genome contains 3 billion base pairs. Basically, it is a letter ATGC. An important goal of human genetic research is to detect the genetic basis of human diseases or traits. Many of you might have heard about GWAS. GWAS is short of genome-wide associated studies, and the GWAS has been successful in detecting genetic variants associated with human diseases in the past 15 years. GWAS is an array-based technology, and one limitation of GWAS is that it only focuses on common variants. The common variance is defined as a genetic variant whose allele difference is rather than 5% or 1% in the study populations. So here is a toy examples. So we assume we have 7 individuals in our data. And now we look at the variant in the gray circle. We could see that there are 3 T and 4 Cs. And hence this variant is a common variant and GWAS data might cover it. So although the GWAS has detected thousands of common variants associated with human diseases or traits, these common variants only explains a small fraction of heritability. A recent study indicates that these missing heritabilities might be accounted for the rare variant. And also the majority of the variants in the human genomes are rare variants. The current biobank level GWAS has hundreds of thousands of individuals. For example, the sample size of the UK biobank GWAS data is 500,000. However, the common variants covered by this large GWAS is still less than 10% in the human genome. The remaining 90% of the variants in the human genome are rare variants. Recent studies also indicate that rare variants have larger effect sizes and are more likely to cause diseases. Researchers also found that coded proteins of rare variants are more likely to be drug targets. So to study the effect of rare variants, a rapidly increasing number of whole genome sequencing studies are being conducted in recent years. For example, the genome sequencing project of the National Human Genome Research Institute and the Transomics for Precision Medicine program of the National Heart, Lung, and Blood Institute, and also the UK Biobank whole genome sequencing data. Different from GWAS, by doing the whole genome sequencing, we would be able to cover every position of the human genome, meaning every 3 billion base pairs. So now we look back to our talk examples in the whole genome sequencing data. We will not only include the information of the common variants in the gray circle, but might additionally detect 5 rare variants, which are in the orange circle. So what does the whole genome sequencing data looks like? I will use the TOPMed freeze 8 data as an illustration. So there are 140,000 individuals in the TOPMed freeze 8 data, and among them there are in total 700 million of the variants. So we could see that analyzing the whole genome sequencing data is a large and huge statistical problem. And among these variants, we found that only 2% of these variants are the common variants with a malloc frequency larger than 1%, and the remaining 98% of the variants are all rare variants. We also found that there are 61% of the variants are singletons and doubletons. For each of these singletons and doubletons, only 1 or 2 of the individuals have the different alleles compared to the others. So the genotype matrix of the whole genome sequencing data is very sparse. Then our question is, how could we analyze this huge sparse data set? The classical analysis strategy for GWAS is to perform single variant analysis of each variant and then do the multiple testing adjustment to detect the significant ones. However, these common strategies could not directly apply to rare variant analysis because of the lack of power. So instead of a single variant analysis, variant set analysis have been proposed to joint test multiple variants in a variant set. Instead of test each variant individually, variant set test evaluate the cumulative effect of multiple variants in a variant set. And when there are multiple variants in the variant set associated with human diseases or trait, the variant set test could increase power. However, there are still some questions remained to use the variant set test for rare variant analysis in whole genome sequencing for whole genome sequencing studies. The first one is how we define the variant set in the analysis. The second question is, given there are many variant set tests has been proposed in literature, which one should be used for the rare variant analysis of whole genome sequencing data? The third one is there are many other bioinformatics databases provide the biological information for the variants. Could we further leverage these information to increase the analysis power? The fourth one is that, so given the whole genome sequencing data set is huge, the computation scalability is also very important. So to answer these questions, I will introduce our proposed whole genome sequencing association analysis pipeline, STARR pipeline. So our STARR pipeline only needs the users to input the genotype and the phenotype information as input, and our proposed STARR pipeline will automatically perform the association analysis and summarize and visualize the analysis result. So specifically, so first our STARR pipeline will automatically functionally annotate the whole genome sequencing data set and generate the AGDS file. So here the AGDS file is an all-in-one file, includes both the genotype and annotation information. Our STARR pipeline also provides tools to calculate the GRM. And then in the next step, for the common variants, our STARR pipeline provides the single variant analysis like GWAS. For rare variant analysis, our STARR pipeline provides the variant set analysis and will automatically define the variant set. We consider two strategies to group rare variants. One is gene-centric analysis, and the other one is non-gene-centric analysis. For gene-centric analysis, we focus on the rare variants in or near genes and group the rare variants based on the functional categories. For non-gene-centric analysis, we focus on the variants in the non-coding genome, especially in the intergenic regions, and group the rare variants based on the physical locations. Then for each variant set, a STARR pipeline use the STARR method to incorporate multiple function annotations to further increase the analysis powers. In the final step, our STARR pipeline also provides several analytical follow-ups, including the summarizations and visualizations of the analysis result, and also the conditional analysis to adjust for the known variants. So in next few slides, I will show you some details of the rare variant analysis in our STARR pipeline. First, I will introduce our group strategies of rare variants. As I introduced before, we consider two strategies. One is gene-centric analysis, and the other one is non-gene-centric analysis. For gene-centric analysis, STARR pipeline provides 5 coding masks and 8 non-coding masks. For the coding mask, we group the rare variants for loss of function variants, and destructive missense variants together, missense variants, destructive missense variants only, and the synonymous variant. For the non-coding masks, we provide 8 strategies. First, as to group the rare variants in the promoters and enhancers and overlaid with the cache of the DRS and the DHS set, and we also provide the mask for the UTR regions, the upstream regions, the downstream regions, and also a mask of the NCRA genes. And for the non-gene-centric analysis, we consider both the fixed size sliding window analysis and also the data adaptive size of the dynamic window analysis. For the sliding window procedures, for example, we provide the 2 KB sliding window procedures with 1 KB skip length. For the dynamic window analysis, we provide scan statistics based method scan G-STARR, which could dynamically detect the window sizes and the locations, and also automatically control the genome-wide type and error rate at any given alpha level, for example, 0.05. And after we define the variant set, the STARR pipeline incorporates multiple function annotations to empower the variant set test using our STARR framework. So here is the workflow. So assume now we have D annotations, and each of these annotations represents the different biological functionalities. For example, here we consider the mallory frequencies, the annotation principles to represent the local diversities, and also the APC conservations, and also the APC protein functions. Then we just choose a variant. Then we just choose a rare variant test. Later, I will talk you the details about which test we used. And for that test and for the weight we choose, we could calculate the p-values. So here in this workflow, then we have the D different p-values, P1 to T to PD. Then in the next step, we use the ACAT method, which use the causal combinations to combine these p-values and generate our STARR statistics T-STARR. And the p-values of the STARR statistics P-STARRs could be calculated in this formula. And in the literature, we show that so this calculations was accurate and fast. Then we need to choose a variant to choose a rare variant test for the analysis. In terms of rare variant test, there are 3 representative existing methods: burden test, SCAD, and the ACAT-V. So the burden test assume all the variants are causal in the variant set, and all the effects are in the same directions. In practice, when majority of the variants in the variant set are causal and all the effects in the same directions, burden test works well. For example, when we consider the loss of function variants in the gene-centric coding analysis, burden test always works well. However, when the effects have the different directions of the causal variants, the burden test might lose power, and the SCAD works well in this case. For both burden test and the SCAD, they all assume the signals is dense in the signal regions, while the ACAT-V test works well when the signals is sparse in the signal regions. Then we just proposed our STARR-O test, where the STARR-O test is an omnibus test that combines burden, SCAD, and ACAT-V using multiple annotation ways. By doing so, the STARR-O tests have 2 good properties. First, by incorporating different tests, STARR-O is robust to the sparsities of causal variants and the directions of the effect. Second, by incorporating multiple functional annotations, STARR-O is powerful when any of these functional annotations can pimp out the causal variants. And finally, I will also give you a brief overview of our STARR pipeline again, which provide the scalable integrative inference of the rare variant association analysis. So in the preparation step, the users need to prepare the genotype data and the phenotype data, and our STARR pipeline provides a tool to calculate the GRM. Then in the next step, the STARR pipeline uses the favor database and the two favor annotators to function and annotate the whole genome data set. Then in the third step, our STARR pipeline will automatically define the variant sets for analysis. Here we consider two strategies. The first one is gene-centric analysis, where we group the variants based on the functional categories, and we provide both the coding masks and the non-coding masks. And the second one is a non-gene-centric analysis. We group the variants based on the physical locations, and we provide the fixed size Zilin window procedures and also the dynamic window analysis procedures. And then for each variant set, we just estimate our STARR statistics by incorporating different functional annotations and also 3 different tests. So here we might consider the annotation to represent epigenetics, conservations, and we combine 3 tests: SCAT, Buren, and ACAT-B. Then in the final step, the STARR pipeline will provide the p-values for all the variant tests and do the summarizations and visualizations of the analysis results. And now I will show you an examples of the STARR pipeline. We will apply our STARR pipeline to analyze the TWAS freeze-5 lipids data. We are interested in analyzing the total cholesterol, and the total sample size is around 30,000, and the number of variants is about 255 million of the variants. So here, our STARR pipeline just provide 2 strategies to analyze the rare variant. One is gene-centric analysis, and another one is non-gene-centric analysis. For gene-centric analysis, we provide 5 coding categories, which are listed here, and also 8 non-coding categories, which are listed here. And for the non-gene-centric analysis, we provide the 2 KB sliding procedures and also the dynamic window analysis procedures using our proposed secondary STARR procedure. And here, first, I will show you the gene-centric coding analysis result. On the left-hand side, this is a Manhattan plot. And here we could see that we find many significant associations in some known lipids associated genes. So in total, there are 19 significant coding masks detected at the genome-wide bufferoni-corrected threshold 5 times 10 to the -7th by adjusting for the number of genes and the number of the masks. And then we do the conditional analysis to adjust for the known variants, and we found that so the 16 of the 19 associations remain significant at the same genome-wide significant level, and these results indicate that so we just find some novel rare variants in some known genes. And on the left-hand side, it is a QQ plot, which indicates our procedures well control the tap and error. And then in this slide, we just show the gene-centric non-coding analysis result. On the left-hand side, it is a Manhattan plot, and also we detect many significant associations in or near 2 known genes, LDLR and the ApoEs. But when we adjust for the known variants, none of these associations remain significant. And the QQ plot is in the right-hand side, which indicates our procedures are well controlled tap and error. And this slide just show you the sliding window analysis result. Also, the Manhattan plot in the left-hand side, just to show that we detect many significant associations in or near some known genes. And in total, there are 59 significant sliding windows detected at the genome-wide bufferoni-corrected threshold 1.9 times 10 to the -8. And after we do the conditional analysis to adjust for the known variants, 2 of these 59 associations remain significant. Also means that we detect a novel rare variants in some known genes. And the QQ plot on the right-hand side indicates our procedure well controlled the tap and error. And this slide shows you the dynamic window analysis result. So the dynamic window analysis procedure ScanGSTARR detects 39 significant regions at a genome-wide error rate level 0.05. And note that the locations of these 39 associations covers the significant findings of the Zilin window analysis. And also our ScanGSTARR procedures additionally detect one associations in intergenic regions on chromosome 9 that is missed by the Zilin window analysis. And here is a brief summary. First, it is positions, and in total there are 60 variants, and the genome-wide error rate is around 0.04, with the region-based p-value at 2.5 times 10^-9. And we found that there are no known variants in the 1MB border regions of these findings, which indicates that this one might be a potentially new region. And in this slide, I just show you the computation resources and indicate that our STARR pipeline is resource efficient. Here we consider we used we just to speed up the computations of STARR pipelines in two ways. First, use the sparse genotype matrix of rare variants, and second, use the sparse GRM. And this table just shows the computation cost for STARR pipeline to analyze the TWAS FRIS5 lipid data. So so with the 200 cores, and we can finish the individual analysis, gene-centric coding analysis, gene-centric non-coding analysis, and the Zilin window analysis just in one day. And if we have 800 cores, we can finish the dynamic window analysis in one day. And finally, it is a take-home message. Our STARR pipeline provides a general rare variant associated analysis framework for biobank scale whole genome sequence studies. We boost the analysis power by dynamically incorporating multi-function annotations, and also STARR pipeline performs powerful and resource-efficient computations for biobank scale whole genome sequencing studies. And here are the implementations of our STARR pipeline. So our STARR pipeline could be implemented in both the university-based clusters and also the cloud-based platforms like the Biodata Catalyst 7 Bridges and also the UK Biobank Research Analysis Platform. And finally, this work is a joint work with Xiaoquan and Xihao from Harvard. And I would also like to thank the support for the TWAS lipids working group, as Gina, Pradeep, Jerry, and Kristen. Thanks for your attention.
Thanks so much, Zilin. So questions for Zilin? Eric?
No, sorry, I was just clapping.
I have one question. So in terms of the recommendation, would you recommend using this dynamic window-based strategy over the sliding window-based strategy, because you showed like some improved results with the ScanSTARR method?
Oh yeah, that's a very good question. So first, I want to just as our result. So because the dynamic window analysis use a data adaptive size. Compare with the Zilin windows use a fixed size. So the dynamic window analysis could increase the power. But when you look at the computation burden, so the dynamic window analysis nearly use 4 times for the computation sources compared to the Zilin window analysis. So I think it is a trade-off. So if you are more care about the power, I recommend you use the dynamic window analysis. But if you are more care about the computation resources, I recommend you to use the Zilin window analysis. So our STARR pipeline provides both the procedures for the users to use.
Any other questions?
I have a quick question. So of course, a lot of the reason that we're interested in rare variants is because they can have much larger effect sizes than common variants. And I guess my question is whether it's possible to get an effect size estimate from something like one of these sophisticated sliding window approaches.
Yeah, that's a very good question, because for the burden test, we just collapse the rare variants adjusted into some kind of one pseudo variants. So we could get the effect size for the burden test, but currently I think there is no approach to calculate the effect size for the SCAT. So there is also an open questions in the field, and we are also currently working on developing some effect size estimations for the SCAT test.
Got it, thanks.
Any other questions? Okay, thanks, Zilin. Thanks for the great talk. We will proceed to the next speaker. So Eric Van Buren is the next speaker. So he is a postdoctoral fellow in Xiaoquan Lin's group at Harvard School of Public Health, and he has pioneered a lot of analysis for IGVF. So Eric, over to you.
Thanks very much. So I'm glad to be following Zilin because I think there's a lot of synergy in the work that we're doing. And I think that I'll try not to be too repetitive, but hopefully seeing sort of some of the same things, at least in the introduction twice, will sort of help people to understand the big picture. So today, broadly, I'll be talking about an extension of the STARR method to incorporate single cell data. And so for today, I sort of titled it Cell-Type-Specific Epigenetics, just because that's sort of all that I'm going to be incorporating today. And so to start, I'll give a little bit of background. I think, again, Zilin probably did a better job than I can about this, but just to sort of emphasize what is important, I think, not only for the cellSTARR method, but maybe even for the IGVF consortium too, how we can leverage rare variant testing and whole genome sequencing data. And then I'll say a little bit about STARR, but again, I think that was kind of covered. And then I'll try to talk about my method cellSTARR, which is sort of supposed to help in non-coding regions. And I'll try to sort of get to why we feel that that's the case. So I think that in case anyone here is not familiar with whole genome sequencing data, I think one of the important things, I sort of like this slide that Xiaoquan made a couple of years ago, but I think one of the important things is that the sample sizes of these data sets are very large. UK Biobank, TopMed is one Zilin mentioned, but we have hundreds of thousands of individuals, and depending on the phenotype, you might have the phenotype in multiple studies. And if you have multiple studies, you might have even close to 1 million samples in the near future for some phenotypes. And so it's a very exciting opportunity because we really have large sample sizes, we have diverse ancestries, and we really can sort of fully study the role that genetic variation plays in the human genome, I think, using these whole genome sequencing data sets and biobank data sets. Today, I'll be talking about TopMed lipids phenotypes again, and they'll actually be from a sort of more current release called Freeze8 that has about 60,000 people, just to give you some idea. And so I think Zilin mentioned this, but just to sort of reiterate that in these whole genome sequencing data sets, what we get out of them are hundreds of millions of rare variants. Most of them are non-coding, and most of them are allele frequency well less than 1%. And even, as he said, singletons, doubletons. You can think of sort of singleton, doubleton, and there are sort of many more columns in this table that are missing to get up to this point here. And, you know, again, most of the variants are non-coding. You know, I sort of have synonymous, missense, and stop gain here. And I think there's a real interest in understanding the associations of these rare variants with a number of phenotypes. And again, as was sort of mentioned in the previous talk, I think that GWAS, the typical approach, is single variant analyses. And I think these are well powered and they sort of have good performance. But when we're talking about rare variant testing, you really have to be a little bit smarter in how you test rare variants. And so what I'm going to focus on today is gene-centric association testing. And I'll sort of get to a little bit how I'm going to modify this, but for today, just gene-centric, and again, just non-coding, but I'll explain why. And so again, I feel that the previous talk was sort of a very nice lead into my talk. STARR was a method that was published in Nature Genetics in 2020. And STARR Pipeline, which Zilin just talked about, was published, I think, within the last month in Nature Methods. But just to emphasize, again, that if you're familiar with the SCAT approach or the burden approach, all the approaches that he talked about and that I'm talking about are sort of building on these approaches. And this figure is very similar to what was showed in the previous talk, but basically the idea behind the STARR method is to say, well, we have many rare variants, and we also independently have a collection of functional annotations of these variants. And so sort of as an aside, in the camp call today, I'll be talking a little bit about this as well, but our portal, which is called Favor, has annotations for every single nucleotide variant and all observed TopMed indels. And we have a very large collection of these functional annotations. And I think the innovation of the STARR method is using these annotations in constructing gene-centric rare variant association tests. And so just to sort of say quickly, you can imagine, for example, if you're interested in loss of function variants within a given gene, if you have reliable annotations about things like conservation, epigenetics, protein scores, selection, et cetera, you can weight the variants that have more evidence of functionality based on these annotations that we've curated. And it turns out that one of the challenges that I think our lab is sort of working from a couple of directions to address is that STARR and STARR pipeline have very few significant non-coding associations, and that's true particularly after you do conditional analysis. And so I put this table up here, and this is actually from a sort of previous version of the TOPMed data, but what all it's showing is for some different lipids related phenotypes, which we sort of use as a good model, the specific genes that have significant associations in each of the functional categories. And the point of this table is just to show that for promoter and enhancer, we don't see the same number of genes that we would expect to see for cholesterol, things like APOE, APOB, NPC1L1, and for HDL, ABCA1, APOC3, etc. And I think that one reason for that is we mentioned that rare variants likely or sort of maybe often have larger effect sizes than common variants, but I think the problem obviously is that non-coding variants are going to have smaller effect sizes than coding variants. And so I think that to start this project, we say, well, we want to improve testing in non-coding regions, and how can we do that? And I think one place that we started with is saying, well, let's be very precise. We'll test only candidate promoter enhancer regions. And among the many definitions of CCREs that you can think of, we actually chose to use ENCODE version 3. And if people have questions, I can sort of talk more about that decision process. But I think the sort of important point to make is just that we want to improve performance in non-coding regions. And I think it's a very natural place at which to insert cell type specific information. There are a number of citations, I put some here, that show that many enhancer and promoter regions show cell type specificity. And so the sort of question that I have been working on since I joined the lab is just, can we improve our ability to detect rare variant associations in promoter enhancer regions through integrating single cell data? And that's what I'm sort of, after all that, that's what I'm here to talk about today, is called cellSTARR. And again, it's sort of STARR plus single cell data. And so I'll sort of go through this, I think, as slowly as I can, but there are two different things that we're really doing to try to improve the discovery in CCREs. The first thing is to integrate single-cell ATAC-seq data from the Catalyst resource. This is a very useful resource of single-cell ATAC-seq data published by Bing Ren's lab. And I think what's very useful about it is that they have over 100 cell types available. So we can test a wide variety of phenotypes. For today, I'm really only talking about lipids, but we can test a wide variety of phenotypes. Any phenotype in which you know or have some idea of what ought to be the relevant cell type or cell types. Hopefully we have sort of matching single cell ATAC-seq data for that. And the idea here is that we can capture cell type specific variation in chromatin accessibility of CCREs. And so I think I sort of maybe I'm abusing the term cell type specific a little bit, but let me just give an example. LDL cholesterol, which I'll show in a minute, we know that one of the most relevant cell types for LDL cholesterol levels is hepatocyte. And what you can think of doing is saying, well, if ENCODE provides us with 10 enhancers, we can look and say which of these enhancers seems to show high or higher chromatin accessibility in hepatocytes. And with that, you can test only the variants from, say, the 5 enhancers that have high chromatin accessibility. And you can actually do sort of an additional step, which is to say, well, of the enhancers that we're testing, you can weight the variants using the STARR framework based on the strength of the signal coming from the Catalyst data. And so this, I think, is a very powerful approach. I hope, and I have this sort of at the end in discussion, but I hope in the future that as more single cell epigenomic data becomes available, that you could incorporate this into this framework sort of very naturally. The other thing that I sort of wanted to mention, but wanted to skip over unless people are of interest for today's discussion, is just that it is a real challenge to link enhancers and promoters to their target genes. So even if you trust that ENCODE enhancers and promoters are very reliable, there are many different approaches to link elements to genes. And what we found is that different approaches can sort of give differing performance. So what we have chosen to do is basically aggregate or combine many different approaches: distance-based intervals, activity by contact, which my impression is that this is sort of becoming the most popular approach, epimap, and screen. And I have a plot that sort of shows, I think, visually how we're trying to combine these things. But really the point in selecting these methods is just that they all represent a little bit different of an approach, and they all use slightly different data to do it. You know, EQTL data versus imputed epigenetics and gene expression versus sort of enhancer promoter contacts. And for now, we're using non-tissue specific elements and links, and this is something that through collaboration with IGVF, we sort of hope to change in the future. And so for today, I'll show some results for LDL cholesterol only, where as I said, you know, we have a number of phenotypes we're looking at, and I think other lipids phenotypes show similar performance. But just to add that I will be showing conditional results. So like Zilin mentioned that in their analyses, many of the enhancer promoter regions sort of do not pass the Bonferroni adjustment conditionally. We've observed something similar, but I will show conditional results. And I think the real opportunity and challenge of this method is sort of which cell types to include. And so I sort of manually curated and pre-specified lipid cell types and a sort of set of unrelated cell types for comparison. And so I put them here, but basically what we're trying to do is just say, as a proof of concept, what cell types should be the most relevant to lipids levels. And we sort of took them from liver, fat, and small intestine. And then for comparison, you say, well, cell types from the heart and brain that should have nothing to do with cholesterol synthesis, transport, etc. Okay. This is a plot that I think really sort of shows visually what the method is doing. So the y-axis is showing the minus log 10 p-value, and the x-axis is showing each of the different gene links that we have. So I mentioned on the previous slide that we have distance intervals. This is actually contained within the gene. This is from 0 to 50 kb, 50 to 100, et cetera, all the way out to 250. And then there's one for epimap, ABC, screen, EQTL, and screen 3D. And as I mentioned, you know, we've seen that depending on the gene and depending on the cell type, that you can see wildly different performances. So what we have chosen to do is use a method called the ACAP method to combine these p-values. It performs pretty similarly to minimum p-value, but statistically, I think it has better properties. And I put this plot up here because APOE is one of the sort of canonical lipids genes. And I just wanted to show that there are differences between the cell types in terms of whether or not you see an association. So like there is an element within the APOE gene. It's still non-coding, but it's sort of between the transcription start and end site that shows association, but this element only shows high ATAC-seq signal in these 4 cell types here. And these actually happen to be the lipid cell types here. And it shows sort of, it does not show high enough signal to be included in the non-lipid cell type regions. So when you combine these p-values, you can see that the signal is sort of flat except here. And so, you know, for our purposes, we sort of find associations in these 4 cell types, but not in these 4 cell types here. And so basically you can picture that each gene enhancer promoter, we produce a plot that looks something like this. And what you can do is you can say, well, which cell type produces the most discoveries at varying significance thresholds? And that's actually what I've sort of chosen to plot here just to sort of demonstrate. So the y-axis is showing the number of promoter enhancer discoveries. The x-axis is showing the conditional p-value threshold. And obviously you can see the size of the threshold that we've sort of chosen to consider, but the point of this plot is really to compare the cell types to one another, not necessarily to validate an individual discovery. And so I think what this plot is sort of showing is that the 3 cell type sort of tissues and their constituent cell types that we presupposed would be the most relevant to lipids, that's liver, adipose, and intestine, show more discoveries using this framework than heart and brain. And I think we were sort of very encouraged by this because, again, it sort of matches biological intuition. And one comment that we received that I wanted to address briefly is saying, well, it's nice that you're including single cell data, but researchers might prefer to include a sort of tissue level assay anyways. So what happens if you include that as well? In other words, is this difference sort of made up for by saying, even if my single cell data doesn't capture it, will the tissue level data capture it? And I think here, we were still sort of a little encouraged in comparing the cell types and tissues together, because liver still sort of tends to show the most association, which again, I think biologically, in particular from hepatocytes, that's a result that we would expect to find here. Actually, I'll sort of skip over that one. And so good, I'm at about 15 minutes. I would love to hear if anyone has any thoughts or comments on how we can improve this method, but I think that I had a few things that I wanted to point out in terms of how this method can sort of link well with IGVF. I think there's a lot of relevant working groups, focus groups. I think in particular, the ability to get cell type specific epigenetic data, cell type specific elements, cell type specific links would really help sort of the method going forward. And I think that there are a lot of methods that sort of do the reverse of our situation. Our method is designed to take cell types that we have sort of pre-specified as relevant to lipids and look for association. But there are a lot of other methods, including SC-Linker by Kushal and others, SC-DRS and SC-GWAS, that are sort of attempting to integrate association results with single cell data in order to identify cell types that are most relevant to the disease. So our hope in terms of methodology is that we can synergize these methods to find the cell types for a given phenotype that's the most relevant, and then test those cell types using our framework. And then I think sort of quick things that people might wonder about is, well, we have not incorporated single cell RNA sequencing information yet. This is something that we actually have some ideas for, but it's actually a little bit more challenging to do, I think, than single cell ATAC-seq data. And then we also have some other extensions sort of trying to leverage the fact that IGVF is going to generate a wide variety of data from diverse populations. And so we're hoping to extend this to incorporate that as well. And so with that, there have been many people that have helped with this work. And again, I would be happy to take any of your questions. Thank you.
Thanks, Eric. So we open the floor to questions. I think, Lexi, you have a question?
Oh, I see it. Okay. The question is, do you see similar performance when using cell lines instead of primary cells? The answer to that is we really have not evaluated that. And I think that's a great idea. I think one of the reasons we didn't really get to this yet is just that because we're studying cholesterol, that's sort of the phenotype that our lab sort of prefers to analyze first, because we sort of understand it well. I think there's not as much we sort of feel like we don't need to use cell lines, but I think that would be a great idea. I think we could try like HepG2 data, or, you know, as you said here, if we were analyzing some phenotype that neurons is relevant, it would be interesting to see if we would see differences using cell lines. So I appreciate that question.
Thanks. Any other questions?
Hey, Eric, I have a follow-up question. So your method is leveraging all these different annotations in the non-coding genome to map these variant associations. Are you able to sort of infer the contribution of each of these annotations to finding new associations? And sort of, if so, maybe what have you found has been the most informative of sort of mapping new associations?
Yeah, that is a great question. And that's something that we've received a lot of feedback over the years saying, well, you know, I can tell you, you know, enhancers linked to APOE are associated with cholesterol levels. But we've received a lot of feedback of what you're saying is that we use these annotations, but the answer to the question is we really do not do the kind of analysis that you're talking about, where we say, you know, if a particular variant is highly associated, what makes this variant special? This is basically your question, right? Is it its epigenetic profile? Is it like its you know, conservation? We really have not looked at that too much. And I think in answer to your question, what we tend to find, I think, is that for many of these associations that there might be a small number of variants that have high association, so like in enhancer regions and probably in promoter regions too. But we really have not done that. And I think that's a good idea in terms of that would tie together well with the consortium's goals of saying, trying to link each variant to gene or sort of to disease and then sort of infer its function from that. So I appreciate that question, but basically we really have not done too much of that yet.
Yeah. Oh, thanks. Curious, are you also limited by sort of just like selective pressures? Like, do you see like many enhancers with no genetic variation, and so you can't sort of map them at all?
The answer to your question is yes, you would see some. I think if your question is about selection, I think that's a good question. So obviously, like if I go back, sort of here, like this method is sort of pretty similar here. What you can think of the cellSTARR is doing is basically saying, instead of this promoter set is composed only of elements that are sort of linked to this particular gene. And I think in order for us to test, there has to be variation, but also it has to be rare variation, right? Like this framework, we're not analyzing common variants at all. I think I should have made that more clear, that if a variant or region has a minor allele frequency of greater than, say, 1%, we don't consider it at all. The framework that Zilin presented sort of it would be more appropriate for that, because it will separately sort of for you test common variants and then separately test rare variants for you. But as I said, here, like we see many rare variants, hundreds of millions, and certainly the more individuals you sequence, the more likely it is that a position will have some variation in a given enhancer, and then if it does, it will be sort of included in our association analysis. So
Cool. Thanks so much.
Does that answer your question?
Yeah.
Okay.
Any other questions? I see there is a question in the chat by Lorenzo. I missed the references for the cellSTARR.
I see that. We're hoping to have a preprint very soon. This slide deck is in the Google Drive, and it has the citations for STARR, and I think STARR pipeline is in here as well. But this work, actually, we're hoping to have a preprint out very soon for it.
So is it the case that the cellSTARR is also going to be part of the STARR pipeline?
I think eventually that would be a great goal. We actually have not sort of talked about it. I think eventually what we would like is to incorporate it that way, because as Zilin mentioned, it's very useful for researchers because they can run these analyses on cloud platforms. And the single cell ATAC-seq data in terms of like data size is not the way that we're using it. We don't need raw reads. We just need basically bigwig files. So I think eventually that would be a goal. And I think eventually that should be doable.
And in general, how does this method scale with respect to the number of annotations and features that you use? Like, because if we start to generate a lot of functional data for a particular, say, cell types or tissues that are of interest, like could there be issues with scaling, or are there some solutions to that?
I think in terms of the number of annotations you use, the method scales very well. I think the only challenge with scalability, if Zilin has other thoughts, he can comment on sort of the STARR family's computational performance, but the more individuals you include, of course, the longer it takes. And the more variants you include, the longer it takes, but I think it's very efficient. And I think especially given the number of variants that we're talking about from ENCODE version 3 enhancers and promoters, it's not, what I'm saying is if you're limiting yourself to only enhancers and promoters as annotated by ENCODE, no matter how many annotations you include, I think it will scale just fine. But if you started trying to test like every non-coding position somehow, that's when you need some of these sliding window approaches or dynamic window approaches, because you have to limit the number of regions that you test. Do you agree with that, Zilin?
Yes, and compare with the sliding window analysis and the dynamic window analysis, I think the gene-centric analysis, as even for the non-coding regions, it is pretty resource efficient.
Yeah,
and it do not require, because you are only testing about tens of thousands of the genes. But if you are considering of the sliding windows or the dynamic window analysis, you are testing for millions of the windows, so they are very different. Yeah. Yeah.
Great.
And and for the and for the basic for the computation course of the eight basic masks provided in the STARR pipeline, you could also find in our GitHub. We just have a benchmark of the computation resources and also the cost for the gene-centric analysis in the cloud-based platform.
Thanks, Zilin. Any other questions? Okay, thanks so much, Eric, for the great
Thank you.
talk. So we'll move on to the next speaker. Luke O'Connor is our next speaker. So Luke is a Smith Fellow in Broad Institute, and his lab mainly works on developing statistical methods to understand the genetic architecture of common and rare diseases. And today, I think he'll be speaking about Bardian heritability regression. Luke, you are muted.
Thanks, Kushal, for the introduction and for the invitation to speak. So I'll be telling you guys about the work that we've done on the heritability and genetic architecture of rare coding variation using exome sequencing data. We have a preprint out on medRxiv describing this work, including a lot of findings that I'm not going to have time to get to. So I do encourage you to check that out. All this work was led by Dan Weiner and Ajay Nadig, who are MD PhD students in my lab and co-first authors on this preprint. And they're really excellent. And I just want to start by acknowledging the hard work they put into this project. The data that we use is mostly from GWAS, which is a UK biobank-based exome sequencing resource developed by Konrad Karakowski and Ben Neal's lab. And if you're not familiar with GWAS, they have a great browser, just gwas.org, based on like the Nomad browser, and just a fantastic resource for querying exome sequencing results from UK Biobank. Today, I'm going to be telling you about a method that we developed to quantify heritability and genetic architecture of rare coding variation. So I'll just start with a quick introduction to common and rare variation, followed by a description of the method and the quantity that we're estimating. And then I'll tell you about some results, the heritability explained by rare coding variants, as well as other things about their genetic architecture and how their genetic architecture compares with common variants. So in GWAS, of course, there have been these two sort of complementary styles of analysis. One involves analyzing individually significant loci and identifying the mechanisms and causal genes behind those loci, as well as things like Mendelian randomization. The other style of analysis involves analyzing common variant architecture genome-wide. And this approach has also been very productive at producing biological insights, including especially with heritability partitioning approaches, looking at cell types, looking at regulatory annotations, and also looking at genetic correlations among different traits. For rare variants, of course, sequencing studies have been very successful at identifying individual significant genes. This is a figure from the schema paper, 2022, where they found 10 significant genes for schizophrenia with really large effect sizes. So these genes here are genes where loss of function variants have really serious implications for patients carrying them. And there have been other papers as well, especially coming out of UK Biobank with many significant genes for complex traits. We know a lot less about rare variant architecture genome-wide. There have been some attempts to characterize rare variant heritability, including especially this paper from Weinstein et al. 2022. But existing heritability estimates are really noisy, and more generally, beyond sort of the whole genome heritability, we really know a lot less about rare variant architecture and how it compares with common variants. So we don't know how much heritability is explained. We don't know whether rare variants are as polygenic as common variants. Of course, polygenicity has been a big challenge in GWAS. And we don't know whether common and rare variants are enriched in the same cell types and sets of genes. We don't know whether they have genetic correlations across the same pairs of traits. So we developed a method called burden heritability regression to estimate the heritability explained by rare coding variants. The motivation is similar to if you were here, I mean, the talks, the last couple of talks outlined why we need to do this type of thing. Most rare variants are observed only once or twice in a sequencing study. They're singletons or doubletons, motivating the use of gene-wise burden tests. And we define burden heritability as the proportion of phenotypic variants explained by the gene-wise burden of minor alleles. So it could be loss of function alleles, could be rare missense alleles. This cartoon is showing 3 genes and the effect sizes of different variants within the same gene. So we expect that loss of function variants within the same gene are going to have identical functional consequences and therefore identical phenotypic effects. Missense variants, we expect to have heterogeneous functional consequences and different phenotypic effects. Synonymous variants, of course, we expect to have little or no effects. And the implication of that is that for loss of function variants, the burden heritability is expected to be approximately equal to the loss of function heritability. By the way, there's some background noise. If someone, if everyone could just check if you're muted, please. For missense variants, we expect that the burden heritability is less than the missense heritability due to this functional heterogeneity that I was just talking about. In particular, like, you know, if half of the missense variants that you include in your burden mask are damaging and half of them are null, then the burden heritability will be reduced by half compared with the total missense heritability. So that's both an important limitation of what we're estimating. Like the burden heritability is smaller than the total heritability for missense variants, and it's also sort of the key choice that makes estimation tractable. So that's what we're estimating. The way we're estimating it is using this sort of intuition. For a non-null gene, the burden statistic, so burden statistic, I mean like the correlation between the gene burden and the phenotype, the burden statistic will increase with the combined allele frequency as you sort of add more variants to that gene. So all the variants have the same effect, and as you add more variants, increases the correlation between the burden and the phenotype, because they're all sort of pointing in the same direction. Whereas if you have a null gene, what you're aggregating is kind of independent or uncorrelated noise between those variants. So as you add more variants and you increase the combined allele frequency, the burden statistics do not increase, or they're not expected to, because these noise and like confounding effects due to population stratification should be uncorrelated between different variants in the same gene. And when we look at real data, we see that real burden statistics do show the expected pattern. So what I'm showing here is these are real burden, gene burden chi-squared statistics for allele cholesterol levels. I've binned genes by their burden score, which is closely related to the combined allele frequency, and then taken the average gene burden chi-squared statistic within each bin for synonymous variants and for loss of function variants. For synonymous variants, where we're aggregating uncorrelated noise and confounding, there's no correlation between the burden score and the average burden chi-squared statistic. For loss of function variants, where we do expect that for some subset of these genes, there's like a true non-zero effect, genes with a larger burden score on average have larger gene burden chi-squared statistics, and there's this linear relationship. Now, if you're familiar with LD score regression, you'll sort of recognize this type of effect as being just kind of like a regression estimator where the slope gives you the burden heritability or is proportional to it, and the intercept is related to the amount of confounding. And that's exactly what we're doing, is we're regressing these gene burden chi-squared statistics on these burden scores. And the sort of regression equation is here. On the left-hand side, it's the association statistic. On the right-hand side are 2 terms. The red term quantifies the burden heritability. The blue term quantifies a few things. Most importantly, just sampling variation. 1 over n is the amount of sampling variance in the sample correlation, and A is some amount of confounding due to population stratification or something else. D here is some overdispersion effects, which don't seem to be very important. We evaluated this estimator in extensive simulations. I'm just showing you one of them. So we used sort of a realistic simulation setup with realistic sort of sparse distribution of effect sizes, realistic sample size and number of genes. Lots of these simulations do include population stratification and negative selection. So, variants with a larger effect at lower allele frequencies. And we get unbiased estimates of the burden heritability, both when it's 0 and when it's, this is 0.5%. At smaller sample size, this is at like 5 times smaller sample size than we have in UK Biobank. These estimates get very noisy, but they remain unbiased. And we also get good estimates for higher frequency variants. These are not common variants. These are variants that are more like 1 in 1,000 as opposed to 1 in 100,000. Now I'll tell you about some results. And again, all of the work that I'm going to show you was done by Dan Weiner and Ajay Nadig. We analyzed GWAS, gene level association statistics, burden statistics. The data looks like this. We have ultra rare variants at allele frequencies less than 10 to the -5. So that's like single digit allele count in UK Biobank, as well as rare variants with frequencies up to 10 to the -3. We broke those variants down into synonymous variants and missense variants that are predicted to be benign by polyphen-2, missense variants that are predicted damaging by polyphen-2, and predicted loss of function variants. So just notice there's much fewer predicted loss of function variants than there are missense and synonymous variants. We analyzed, of course, we analyzed 22 traits and a sample size of up to 400,000. We find that burden heritability on aggregate is 1.3 of phenotypic variance. This is 1.3% averaging across the 22 traits, and aggregating rare and ultra rare loss of function and missense damaging variants. This 1.3%, of course, is concentrated in loss of function variants, so most of it is in these the loss of function variants in red, even though that's a minority of the variants and alleles. And of course, reassuringly, it's absent in synonymous variants as well as in predicted missense benign variants, indicating that BHR successfully controls for confounders. We find that, of course, so this number of 1.3% is much smaller than the common variant heritability. And I think this is expected. So for these same traits, the average common variant heritability is about 10 times larger. So this is a common variant heritability estimated using LD score regression on the x-axis. And you can see the x-axis goes up to 0.5. The y-axis is the total burden heritability. It goes up to about 0.04. So this means that the burden heritability due to rare coding variation is less than 10% of the total SNP heritability. I'm actually going to skip this. I mentioned about confounding earlier. So the intercept of the BHR intercept quantifies the amount of confounding in your burden statistics. And so we looked at the intercept for synonymous variants and for loss of function variants. We see that the average intercept is between like 1 and 1.1. So 1 means there's no confounding here. This is like the average like chi-squared statistic. So there's something like 5% of the variance in your z-scores will be explained by confounding, is what these numbers mean. And that's a number that's like important if you aggregate across the genome, but it's not going to lead to a lot of false positives. So it does look like that 5% is about normally distributed, right? So it's like the confounding effects, just like the sampling variation, are roughly normally distributed. And you can see that by looking at lambda GC. So lambda GC will agree with the intercept for synonymous variants, which don't have any true signal, if the confounding effects are approximately normally distributed. And that is what we observe. So we think that this is a sort of a phenomenon sort of spread out across the genome, which is good because it doesn't mean that it means it's not going to give you lots of false positives. So we estimate that the inflation in the false positive rates due to this amount of inflation is very small. So very small amount of inflation in false positive rates, but important to account for when estimating things like the heritability. Okay, and now I'll tell you about some results that I think are particularly interesting related to polygenicity and the impact of negative selection on genetic architecture. So negative selection, we know, strongly affects common variant genetic architecture. This is a paper that I really like from 2017, looking at LD-dependent architecture. Variants with less LD have much larger effect sizes, and that's mediated by things like allele age that are related to the effect of negative selection. This paper from 2019 looks at allele frequency dependent genetic architecture. Of course, low frequency variants have larger effect sizes than common variants, and rare variants have much larger effect sizes still. In 2019, we published this paper proposing that negative selection actually explains the extreme polygenicity of complex traits. So under the flattening hypothesis that we proposed, the reason that common variant architecture is so polygenic is not that, like, thousands of genes and regions of the genome are equally biologically important for a trait. Rather, the reason is that actually a small number of genes in the region of the genome are like much more important than others, but we don't see them due to negative selection. So here in gray is kind of the distribution of de novo effect sizes, if you could see the effects of like newly arising mutations. And that distribution is dominated by this large effect gene. But when we look at common variants, we're seeing this sort of narrow orange slice along the bottom. These are variants whose effect sizes were small enough to become common. And if you look at the distribution of heritability among common variants, it's spread much more evenly across the genome, across sort of large and small effect genes alike, due to the effect of negative selection. So the key prediction here was that in contrast with common variants, which are this orange slice, rare variants would be much less polygenic, more similar to the gray distribution, because even though they still have a threshold, that threshold is like much higher. So it should be possible for the large effect genes to stand out. There should be a much smaller number of large effect genes, and we should see that in reduced polygenicity for rare variants versus common. And this is indeed what we observed. So on the right here, I'm showing you the heritability explained by individual common variant loci. So each box here is like one locus. The bar is like the fraction of common variant heritability explained by all genome-wide significant loci. And of course, these boxes are very, very small. It requires a very large number of common variant loci to add up to the total common variant heritability. Whereas for rare variants, these individual boxes are much larger, and individual exome-wide significant genes frequently explain more than 1% or even like more than 5 or 10% of the total burden heritability. This is like a large qualitative difference. It's not explained by things like loci versus genes or anything like that. And the idea that this is related to negative selection is supported by the fact that rare variants are much more enriched in constrained genes. So constrained genes, these are like the 20% of genes with that are most intolerant to loss of function variation from NOMAD, and their common variant heritability enrichments. I should say these enrichments are estimated using this method that we developed. It's called AMM, it's the abstract mediation model. Sorry, my light just turned off. This method of estimating common variant enrichments accounts for uncertainty in which SNPs map to which genes. So this average 2x common variant heritability enrichment is not diluted by the fact that we don't know which SNPs map to which genes. It's not diluted. Anyway, the rare variant heritability enrichments are much larger. So what that indicates is that constrained genes, they're probably like, it's not like they're only biologically important for rare variants, not for common variants. No, they're equally, they're probably like equally biologically important for like coding and non-coding stuff. But it's just the common variant signal is strongly flattened by negative selection, and the rare variant signal is not. Now, not everything is different between common and rare variants. We looked at some cell-type-specific differentially expressed gene sets. So these are gene sets from Finucane et al. 2018. These are mostly tissue differentially expressed genes, as well as cell-type specific gene sets from Kushal and others. And we actually find, looking at these gene sets, that the common and rare variant heritability enrichments line up quite well. So, for example, genes expressed in liver are strongly enriched for LDL cholesterol heritability, and they're about equally enriched for common variant and for rare variant heritability. And that's true roughly across the board here. We also observe similar common versus rare variant genetic correlations across traits. So here on the upper triangle, I'm showing you the common variant genetic correlations estimated using LD score regression. And on the lower triangle, I'm showing the rare variant correlations estimated using the cross-trait version of BHR. And we see genetic correlations between most of the same pairs of traits. On average, this is the common variant correlation versus the burden genetic correlation. On average, these rare variant genetic correlations are a bit stronger, but they're concordant in the sense that you get the same trait pairs. So now I'll wrap up. We found that rare coding variation does not explain that much heritability. The burden heritability of rare coding variants is 1%, 1.3% of phenotypic variants. And on the one hand, that implies that rare coding variants are not going to solve the missing heritability problem, and they're only modestly going to improve the performance of polygenic risk scores. I mean, it's basically like most people just don't have one of these variants, so that puts an upper limit on how much improvement you're going to see in polygenic risk scores. Of course, these variants are very important for the individuals who have them. On the other hand, I'm very optimistic about the biology that we're going to learn from rare coding variants. Our polygenicity results suggest that rare coding variation is going to implicate a tractable number of large effect genes. Like, it's been a huge problem in GWAS that we get so many loci, so many genes, and they have small effects that are hard to interpret. For rare variants, it's not going to be the case. We're going to get really a much more tractable number of these genes, and they're going to have large effect sizes, so hopefully they'll be more biologically interpretable. At the same time, I think these results add to sort of a growing evidence for mechanistic convergence between common and rare variants. And mechanistic convergence is important, again, because so few individuals have rare variants in these genes. And if it weren't the case that you saw this mechanistic convergence, you would worry that biological insights derived from these genes would only be applicable to the individuals who actually carry the mutations. But these sort of early signs of mechanistic convergence, here we're seeing gene sets and the genetic correlations. It's also been observed the level of individual loci and the level of pathways, suggests that actually the sort of pathways and cell types and even individual genes that we identify using rare variants will be applicable to a much larger number of patients, most of whom have elevated common variant risk instead of rare variant risk. And maybe even therapeutics that are developed targeting these genes will be applicable to a large number of patients as well. And I'll just wrap up by saying that BHR software is available on GitHub. This is like, software is still sort of under development, and we definitely be eager to help you implement your own BHR analyses if you're interested in running BHR on your own data. And again, thank you to Dan Weiner and Jana Digg, who did all the work that I showed you, as well as to our other co-authors, especially Conrad, who developed the data, and including as well Kushal. So I'd be happy to take a couple questions.
Thanks so much, Luke. Questions for Luke?
So I have a question. Sorry. So I think I have one question about that. So you see that, so you observed some inflations using the burden test, especially for the ultra rare variants, right? But my question is, did the GWAS use the C-gene test or the C-gene plus test? Because I think there is a recently published Nature Genetics paper just mentioned that if you use the original C-gene test for the burden test, it will have the systematic inflation. So I'm worried about whether this is this is why you observed for the lambda there is a inflation, but it is because of you have the systematic inflation of the C-gene test that you use. But now based on the same author's paper, you need to use a C-gene plus test.
That's a great question, and we should look into that. I'm pretty sure this is based on Sage-Gene, not Sage-Gene plus. So that would be consistent with the findings of that paper. And again, the amount of inflation that we observe, I think, is not like very problematic, especially for like if you're interested in single gene tests. It's not going to give you lots of false positives, but it is it is the kind of thing you have to account for in heritability style analysis. I didn't realize this came out with Sage-Gene plus. So thanks for pointing that out. I'll definitely look for that paper.
Yeah, because I think the same author just published the same paper. They just mentioned that for the ultra rare variant, the original Sage-Gene test have the problem. Yeah.
Andrew?
So this is all done with coding variation. So I'm curious if you wanted to look at rare variants in non-coding regions, particularly like regulatory elements. Let's say we have regions of enhancers. Is it easy to just extend it to that, or are there greater complexities involved?
That's a good question. So I mean, I have a lot of thoughts about this. I'll try to sort of organize them. On the one hand, it's sort of mathematically straightforward to extend BHR to non-coding variation. Like there's no real difference between like burden, like a gene burden, and like an enhancer burden or like a window burden. Like mathematically, there's no like obstacle there. At the same time, I'm not that optimistic that we're going to find a bunch of heritability that way. I like the selection coefficients that you observe in coding and non-coding regions are really different. And probably especially for ultra rare variants, it's hard for me to imagine that we're going to get a bunch of burden heritability looking at ultra rare non-coding variants, especially if you aggregate them in some sort of imperfect way. Like loss function variants are great. You can aggregate them so easily. I'm a little bit more optimistic about variants at intermediate frequencies. So if you're talking about, stuff around 10 to the -3, it's easier to imagine that the non-coding variation is going to explain a chunk of heritability. But at that point, I'm not sure if you need burden heritability regression anymore. If you're talking about 10 to the -3 variants, you might have power for individual variant tests or individual variants heritability estimation methods, like just like something normal like REML. What would make me more optimistic would be if you had extremely good annotations, or even like variant effect predictions. There's been a lot of sort of rapid progress in predicting the effect of variants on epigenetic markers, local epigenetic markers, as well as gene expression in cysts. That's methods like an early method was it called Expecto. There's a method called Informer, and then another method called Say that came out recently. And maybe with a method like that, you could bring the burden heritability much closer to the total heritability for non-coding variants, and then maybe you'd also have power to actually see it. So I don't know. This is like a long. I don't know. This is probably way more than you wanted to hear about non-coding. I have mixed feelings about it, basically.
Yeah, I think one interesting point you brought up was that the burden heritability estimates are way lower than sort of the common SNP heritability, but we're only looking at coding variants. So maybe a natural extension would be, maybe the rest is in rare variants and non-coding regions. And that might more align with, say, Peter Vischer had a paper last year or 2 years ago that sort of implicated that. So curious if any thoughts.
I mean, that's the Weinstein et al. That's the Weinstein et al. Paper, which it was just published. It's been a preprint for maybe even 3 years, but it was just published this year on Nature Genetics. And I mean, so they estimated that all the heritability is all the twin heritability is explained by the combination of common and rare variants, but like their estimate for the contribution of rare variants specifically was not even significantly different from 0. So it's really hard to tell whether what happened was they just had a bunch of extra noise, and the noise brought the estimate higher. So I think the jury's still out on whether rare variants and rare coding variants in particular explain a bunch of missing heritability. I will say that Kushal et al. 2018 Genetics compared common and low frequency variants and the fraction of common versus low frequency heritability that's explained by coding variants. And what they found was that even though coding variants are only like 10% of heritability for common variants, they're much higher, like more like 30% of heritability for low frequency variants at like 0.00, it's like probably like 0.005 to 0.05 was low frequency. And if that trend continues, it's quite plausible that like most of the rare variant heritability, and especially the ultra rare variant heritability, is coding. And I think that's especially true of rare non-coding SNPs. I think that for rare non-coding structural variation, I think it's much more plausible that you have like a lot of large effect sizes and large selection coefficients. For rare non-coding SNPs, I'm not that optimistic about finding lots of heritability.
Okay, thanks so much.
Other questions for Luke? Okay, thanks, Luke, so much for your talk. I'll move on to the next speaker, Mike Love. I think Michael is here. Mike is an associate professor in biostatistics and genetics department at UNC Chapel Hill, and it's great to have him present the last talk of this workshop, which Mike and I co-organized, so it's great. Mike, you were muted.
Of course, thanks. I was just saying thank you, everybody, for staying with it to the end. Yeah, let's see if I can break before the end of the hour. So I'm going to be talking about estimating gene-to-trait effect sizes in multi-signal GWAS loci. This is very much following from what we heard at the very beginning from William and Jean, and also yeah, I had to step out briefly, so I'm going to have to go back and watch the recordings for the middle talks. Maybe there was also some mention there. This is collaborative work that was also done with people at UNC in the biostatistics department and neuroscience center, and ongoing work in collaboration with people in our UNC center, IGVF center. So Zilin Li and Karen Mulkey helped us out in this work, and ongoing work with Sarah Brotman in Karen's group. So I'm going to first just present some terminology. So we're interested in loci that exhibit allelic heterogeneity. And by the way, if you have any clarifying questions, I have the chat window open so I can answer them as they come up. So we're interested in loci that exhibit allelic heterogeneity, or you could say like exhibit an allelic series, where we have, here I'm showing a Manhattan plot with the EQTL minus log p-value for the gene LIPC in liver. And this data has, in this case, this has been clumped. So we have signals where all the purple variants that are highlighted are in LD with the lead purple EQTE SNP in a triangle. And we have low correlation, low LD across these three signals. And if you line it up with HDL GWAS below, so this was described earlier by Jean as a Miami plot, you can see that there's corresponding signal in the GWAS locus and in the GWAS study. And although it doesn't seem like a very significant down here, just the size of the small p-value for these other two green and red signals is kind of like making that not look very big, but it's still a very significant signal, this purple one. And if you're interested in like lining up these lipid traits with EQTL and GWAS, there's a guy, Eric Faumann, who writes these threads about these on Twitter. So if you're interested in like the story of how these metabolic pathways work and how you can use GWAS to figure out metabolism, you can follow him. So just to define some terminology, I'll refer to like a region where there's signal as a locus. It's just a very generic term, but in particular, we can talk about a lead variant for one signal where we would, we have multiple ways of detecting like multiple signal locus. And this other, tower here is indicates a lead variant for another conditionally distinct signal. And William had a plot showing, conditioning is kind of a great way to show that a signal is independent, or I should say conditionally distinct. So if you condition on one, the other goes away, and I'll show an example of that. So this is what conditioning looks like, and this is from a paper from Karen's group. So at the top we have waist tip ratio GWAS minus log 10 p-value, and there are two variants that are highlighted. So in green are variants that are associated with 804. I'll just refer to the last three numbers. And then in purple are variants that are associated or in LD with the variant 377. So if you perform an EQTL, a scan in this region for the gene DGKQ, you get kind of both signals entangled. If you then condition on the variant 377, that drops 377 to a p-value of 1, or cannot be estimated, and all of the variants that are in LD with 377 also drop, because that signal has been explained by the conditional genotype. And then what you can see is the emerging, like you can now more easily identify all the variants from this 804 signal. So this, in this case, would to help colocalize the waist tip ratio signal associated with 804 with this DGKQ signal. So how many genes exhibit this, like where there are multiple conditionally distinct sets of variants associated? And like I said, the gold standard is this conditional analysis, and the estimates, you go for the studies that have large sample size. And so here, a study of peripheral blood with almost 5,000 samples showed maybe a third of genes had more than one conditionally distinct signal. It has a range if you look in GTEx, but importantly, the number of genes with allelic heterogeneity or multiple signals just correlates with number of samples. So that's critical when thinking about GTEx, is that you're discovering, you're not discovering everything. And I just want to emphasize that with a second slide. GTEx is a fantastic resource, but it's not discovering all EQTL. So if you're working with GTEx data, your discoveries are the ones that were able to be detected, but it's not discovering everything. And so an example, like a simulation study from Michael Inouye's lab has a nice figure which helps you think about power. And so GTEx is on the left side of this plot with the sample sizes typically in the range like around 200 for most tissues or less. And so you can just follow this plot along, and looking at the minor allele frequency for the variant, you're just not detecting everything in GTEx. So that's an important consideration. So we can instead, boost by combining studies. So in this case, this is work from Sarah Brotman and Karen Mulkey's group. Ongoing work was presented at ASHG recently. A meta-analysis of EQTLs in adipose, where here Sarah has these columns represent genes with additional signals. So the one column would be just a single signal, or two or three, etc. And the rows represent different studies with their sample sizes, and then a meta-analysis combining these studies together. And in particular, if you look at like this set of percentages here, there are quite a lot of multi-signal loci here. So it's actually more common than not, right? Because the one column has 49% to have multiple signals for the GWAS. And this is just a diagram of those numbers. So the black is the meta-analysis, helping you rise above and detect these multiple signals for EQTL. This is going back to what Jean had introduced earlier today. We can use these multiple signals when we have them to perform Mendelian randomization, and we have a method from our group called Mendelian randomization in the per locus, so MR locus, and its steps are basically to perform ideally conditional analysis to separate signals, so show that they are conditionally distinct. We take the E SNP is the instrumental variable, and then we just bring along the summary statistics for the GWAS. So kind of the input to the method is summary statistics, and for the EQTL and for the GWAS, and then the LD matrices from within these signals. And the next step is colocalization within the signals. We try to find the best candidate variant. We're not trying to find the causal variant, but just a good representative from within the signal. We trim so that there's less than R squared of 0.05 across the signals. And that's because we're not actually modeling the correlation of the instrumental variables. Another thing that Jean mentioned, some methods do that, other methods have to rely on trimming. And then finally, we estimate the slope. What we were interested in is the slope of the gene dosage on how that affects the GWAS trait. We do that with a Bayesian model. So, another thing that Jean mentioned earlier was that not many methods include the instrumental variable standard error. And that's, I think, if there's anything you remember from today, from my session, it's that you really have to include the instrument standard error when you're talking about cis Mendelian randomization. It's non-ignorable. It's much larger than the GWAS standard error. So that goes in here. I'm using beta hat where Jean had the capital gamma hat. But it's the marginal association from the EQTL study for A. A is like the proposed mediator, and B is the downstream GWAS trait. So we include this standard error for the EQTL effect size, so the beta hat A, and PTWAS, which is a method from William Nguyen's group, does something slightly different. So it weights the instruments by the PIP. And so it's kind of like two different approaches, but I think it's very important one way or the other to model. When you're trying to model the mediated effect size, you have to include this uncertainty on the on per signal. So just to show you a diagram of what that looks like, because I'll be showing you some plots applying this to real data. So we have, we're trying to estimate this parameter alpha. So alpha is the it's basically, the underlying association for GWAS regressed on the underlying EQTL effect size. We don't observe those, we observe the beta hats. So we're trying to put these into a Bayesian model. And the blue slope would be the slope alpha. We also get a credible interval for alpha, which is these dashed, like the cone. And then we also estimate sigma, which for us is the dispersion. So how much the multiple signals agree or disagree on the mediated effect size. So for us, the most two interesting parameters here to perform inference on is alpha and sigma. And just to show you like how would that look across different scenarios. So on the left side is when alpha is large, we have a large slope we can detect, and sigma is small. And then in the middle is alpha is large, but now sigma is large. So the multiple signals are not giving a very consistent picture. And I think just looking at these two pictures alone, I think looking at cases where sigma is large, but so we think there's a mediated effect, but there's some disagreement across the multiple signals. I think probably that might indicate that we have a proxy tissue, which is not actually the tissue in which the heritability is mediated. So we get kind of, we're capturing some of it, but then some other of the signals are, you know, either it's like, for example, if there was a point which was like right on the y-axis, on the x-axis, it might indicate an EQTL in this tissue, which has no effect on the trait because it's the wrong tissue. So it's just, you know, there's some amount of sharing, and we're not capturing the right tissue for the study. And then the final plot on the right is the case where there's no mediation. And we've thought about ways of compare, you know, you can't, sigma is on the scale of the GWAS trait, so it's not easy to compare across different studies. So we've thought of a way to normalize it is to divide by some position on the y-axis, and a reasonable position for us would be if you project from the average EQTL effect size, so take the average of the EQTL effects, the betas, and then just project that on the y-axis. So we call this like the normalized allelic spread, and it allows us to compare then across different studies to see, you know, how spread out the signals are. So this is an example of applying this to that meta-analysis in adipose. So this is kind of like one of the very nice looking genes where we have 3 signals, and they're each giving a kind of consistent picture in terms of the effect size and also even in the dosage, right? So they're lined up in terms of their EQTL effect size and their effect size in GWAS. And so we can estimate this slope, which is interpretable as if you were to change the gene expression by 1 standard deviation, what would be the effect on the GWAS trait in terms of the units of the GWAS trait. And here's just a couple more interesting MR plots. So the previous one was PDE3A on HDL. And this was presented by Sarah at ASHG. And then here's some other examples that look nice from our scan. We have LPL on coronary artery disease, GALN T2 on HDL again, and then CCDC 92 on waist hip ratio. So this is like MR in practice, cis-MR in practice. So I just want to mention some limitations. Obviously, there's a lot, you know, the first 2 talks really give a great overview on how difficult this is. And so here, we're not explicitly modeling multiple genes per locus. That's one thing. We're just kind of scanning 1 gene at a time. We're not considering horizontal pleiotropy. So for example, if there were SNPs, which then, you know, which were not just affecting the gene expression, but we're going, you know, through a different route. So we're not, we're kind of assuming the exclusion restriction. We're trimming signals, so we're throwing away, you know, information there instead of modeling when the instruments have high correlation. And so that would just, you know, complicate the Bayesian model, but you do lose it, you know, it's a lot of loss of information when you have to trim a signal. And also, we're assuming here that there's no sample overlap. There's interesting approaches to try to compensate for some amount of sample overlap between the EQTL study and the GWAS. So just to summarize, I think multi-EQTL signal loci are probably the rule, not the exception. And it's just that we've been scratching the surface with most of these tissue-specific studies, EQTL studies, which, you know, it was great, but I'm very excited to see the EQTL, you know, single cell EQTL data sets start increasing sample size. We're probably not going to see a lot of these multi-allelic loci, or sorry, multi-signal loci until we start emerging out of the hundreds of samples. We have to get up into the thousands of samples to start to find these, but they offer a lot of promise. So you just get a lot of extra information about gene to trait effect sizes in, in cells and in tissues, hopefully in organs and tissues. I think I pointed out these models are just starting to scratch the surface. So use of co-data would be very helpful to refine the estimates and to refine, the variance. And I think their perturbations are going to be really exciting to add into these models. So combining perturbations from IGVF with QTL studies. And another thing to note is like mileage varies. So it looks great when you have like a gene that metabolizes something and you have the gene expression as the mediator and the lipid level as the GWAS trait. Clearly there's a continuum of how close the traits are to the mediators. And so we have these great examples for HDL and LDL. And as you get further out, mileage varies, I think. Yeah, so we have, there's an R package for this. Recently, I'll just mention, recently we have a Bayesian model for colocalization, but we've actually just been using eCaviar, which was mentioned in the beginning of the workshop, just for speed, so that we can do this, rapidly scan across, hundreds of genes or thousands of genes. And just to stop here, I'll acknowledge all the people who work on this. So Anshi and Nana were the co-first authors for this paper. Jason Stein was a co-PI on the work. Lots of great feedback from various diverse people and groups, including folks in industry as well as academia. We'll continue to work with Karen and Sarah and Cindy and Michael Gandel on Cindy Wynn and Michael Gandel on neuropsychiatric traits, and thank our funding. This was supported by NIMH predominantly. And then the last slide is, I do have a bunch of papers that I like that are in this space. So there's just like, you could go to town on reading papers for estimating these mediation effects. I'll stop there.
Thanks so much, Mike. Great talk. Questions?
I have a question. Have you looked at, that's a great talk. Have you looked at whether loss of function variants in some of these same genes have effects? I mean, even if they're not, you probably don't need to restrict to like exome-wide significant stuff. Have you looked at whether they have effects that are concordant with the effect sizes that you are estimating?
That's a great question. I would love to, we have not. From looking at the neuropsych, so we've run this for schizophrenia with fetal and adult brain expression as the mediator. And we see genes popping up that already have been known from whole exome. But your question was more about the effect size lining up, and I would love to do that. I think that's like a great question for IGVF is how do mutations that affect protein and their effect sizes line up with these kind of dosage effect sizes and splicing, I guess also splicing of, like how do those effect sizes correlate and what should we expect? Yeah, that's a great question.
Yeah, I mean, for some of those genes, like, you could just, HDL, for example, is in GeneFast. You could just go and, look it up right now.
Yeah, I'll put it on the to-do list. It's a great point. I love to talk effect sizes. I think we need to move beyond the p-value world and into the effect size world.
Other questions? So, Mike, I have a question related to the fact that with the current efforts being also to generate a lot of these disease case control data sets and to generate EQTLs that are more dynamic in nature. And that kind of gives a window into understanding what might be the EQTLs that are changing in their effect sizes between the healthy state and the disease state as well. Do you see a way to kind of pull that information into this?
Yeah, that's another great question. So this doesn't have you know, this model is pretty simple, but one, you know, like it's coded up in Stan, for example, and a reason I like that, I like to, you know, work with those kind of models is because you can quickly, you know, add something. So you could add in like, you know, a split across two types of QTL effect sizes and see like when are the effect sizes mediated through both or only through the, you know, the like treated state or the disease state. But we don't have that yet. I think that would be another like really exciting place for the computational modelers. And I'd love to be involved as well in seeing that. I think it makes sense to think about it like, you know, other ideas I'm just stealing a lot from Gene today are like thinking about it as mixture modeling, like, you know, some set of these variants are mediating, some of them are mediating only in certain contexts and in certain tissues. And so like setting these up as mixture models makes a lot of sense, I think.
Great, thanks. Other questions? Okay, if not, we can thank Mike, and thanks to all the speakers for the great talks throughout day 1 and day 2 of the workshop. And we plan to have the talks uploaded on the IGVF YouTube channel. And also in the meantime, we'll also have it on the Google Drive that is shared with all the IGVF members for that. So if there are any other items, you can let us know, but otherwise we can probably call it a day.
Great. Thanks, everyone.
Thanks so much.
Thank you. Bye.